Hans and I are basically like 50 meters away from each other. If you're still in the office.
Yes, indeed.
Yeah.
50 meters and a few walls.
A few walls, yeah.
We lost one of our speakers, it seems, but hopefully they'll be back.
Okay. So it's one of the first times I will be giving a talk at the neuroscience conference in October, November without the jet lag. Normally it's like for neuroscience meeting and there's always a jet lag involved then, but now I have no problem with that.
No excuses anymore then.
Yeah, that's true. If I'm sleepy, then it cannot be the jet lag.
Tessle did say she might have Wi-Fi issues, so.
Okay. Well, she's the last speaker, so hopefully time to sort it out. Yeah, I'll just keep an eye out for her anyway. Hopefully we can.
Okay.
Right, I'm gonna hop off, or I'll be in back end anyway, but thanks for being here, guys. See you soon.
Thanks, Sean. Okay, welcome everyone to this this session of NeuroMatch 3. My name's Elaine and I'll be introducing the speakers today. So first I just will quickly remind you that if you have any comments or thoughts that you'd like to share on Twitter, to please feel free to do so and use the hashtag NMC3. So we have a fantastic lineup of speakers today. We're going to be learning a lot about visual processing and how it's affected by expertise, cross-spatial scale coherence, and also learning hopefully about prediction in visual motion processing. So this is an interactive session, so there'll be short talks with loads of time for questions. So please do ask lots of questions and we'll get a good discussion going. You can also use the chat to send, to make comments to everybody, and you can vote on what questions you like, you find interesting in the Q&A. And if you'd like to ask your question on screen, please either type on screen beside your question, or you could raise your hand as well. And if you prefer not to, I'm really happy to ask your question for you. Okay, so our first speaker today is Hans Op de Beeck. Hans is a professor in the Faculty of Psychology and Educational Sciences at the University of Leuven. And he's the director of the Department of Brain and Cognition and a faculty member of the Laboratory of Biological Psychology. Hans's lab does some really interesting research investigating the neural basis of visual cognition and learning. And today he's going to talk to us about the effect of expertise on behavioral, neural, and computational representational space. So, thanks very much, Hans.
Yeah. Let me share my screen. Thank you, Elaine, for the very nice introduction. So I will indeed be talking about the effect of expertise, and we did some fMRI to understand how expertise is changing brain function. And to investigate that, we did not look at faces because we are all experts at face recognition, so faces are not really special. And to study how expertise changes brain function, it's good to have people that don't have a certain expertise. So we went for more obscure domains of expertise that not all people have. So these are special domains, and only a minority of us master them, but many of us have such a specialty of some kind, either in a formal system, a formal hobby, for professional reasons, or even something that only you can do. And if you have an expertise of that kind, feel free to also put it in the question and answer part, and then other people can vote for it, and maybe you can be the champion expert of this session. So feel free to do so. So what can we investigate then? Well, like I said, we want to investigate how this changes brain function. Now, previous neuroimaging studies have mostly looked at which regions are involved if you see the category of objects you're an expert for. And one big discussion, for example, has been whether the fusiform face area is involved in certain domains of expertise. And the answer seems to be that it depends. So there are clearly differences between domains of expertise. And one such study was one we did in the lab a few years ago, where we compared bird expertise with mineral expertise and found very different effects in how this expertise changes the neural processing of the objects of expertise. In particular, in visual cortex, in high-level visual cortex. In frontal cortex, there were also very clear changes, but they seem to be more common between the different fields of expertise. Now, the problem is what really matters for expertise is not that you can recognize an object as being part of that domain. For example, a bird expert is not a bird expert because he can tell us that something is a bird, but he can tell us which type of bird it is, which species of bird it is. And that's the base of the behavioral definition of an expert. So that's what we should be looking for in the brain as well. And neuroimaging studies have not done that so much. When they've done it, it's mostly through adaptation studies, where you use the fMRI adaptation as a way to get access to more finer representations. But you don't get much information about the representations themselves and the nature and the geometry of these representations through fMRI adaptation. So we have methods for that, representational methods like representation similarity analysis, using multivoxel pattern analysis, all of that. But it's challenging to look at subordinate difference between stimuli because we know the signal is much weaker. And this is a study or a figure from a study from more than 10 years ago already showing that it's quite tricky to do. Nevertheless, we tried that. We studied expertise in ornithology, in bird watching. We scanned 20 experts and 20 controls. And we used a design with 24 stimuli, all birds, of course. And there was some attempt of us to dissociate the visual characteristics of the stimuli from the more semantic information, in the sense that there were 8 triplets, and within each triplet, the B stimulus was visually quite different from the A stimulus, but yet belonged to the same species. And it was not more different from stimulus C in terms of the visual aspects, yet B and C are from a different species. We first did some behavior, and if you look at the behavioral dissimilarity matrices, you get something like this. So you have experts above, controls below, and we have 2 tasks. And you can see that overall, these representations, these matrices look quite similar. So overall, there's quite some similarity in this representational space. Nevertheless, in the experts, we see some difference depending on the task. So a task difference, which we don't see in the controls. And in particular, in the species task, you see certain kind of groupings that you don't see in the other people. During the scanning, actually, we went for an overall similarity task that doesn't really tap into this specific species aspect of the stimuli. Now, behaviorally, what we also see is that the experts are much more consistent among themselves in the representation. So if we correlate the matrix of an expert with the matrix of another expert, we find a higher correlation than when we do the same thing among controls. So the green bars here are higher, and we see that particularly in the species task, even though it's also significant in the visual task. So let's now go to the neuroimaging part of this study, and then we get results like this one. So we present 24 stimuli, we look at the pattern of activity across voxels, and we correlate this pattern of activity between conditions and themselves, a repeat of the same condition, or between different conditions. And then you get a film matrix like this one. This is the one for frontal cortex, so a big region encompassing all of frontal cortex. And then you see that there is some structure in there, both in controls and experts, but the structure seems to be more pronounced in the experts. And you see a bigger use of the color scale, and the color scale is matched between controls and experts. We did this in 3 large regions of interest, the same region that were used in that previous study on ornithology of 2018. And in early visual cortex, there's a really good match between what we see in controls and experts. In high visual cortex, it doesn't look exactly the same anymore, and then in frontal cortex it's clearly different. So now I'm doing a Rorschach test, and it's like looking at these pictures and seeing what you see in it and what I see in it. Of course, we need to do some statistics in science. So let's see what actually drives this impression of a stronger representation in frontal cortex. The first thing we looked at are, again, these intersubject correlations in the neural dissimilarity matrices. And we again see that these correlations are higher for the experts than for the controls. In particular, in frontal cortex, this effect is very strong. In the high-level visual cortex, it's also there, but it's weaker, but it's also significant. A second effect that we noted in frontal cortex is that the diagonal is most pronounced. And the diagonal means that you compare a condition with itself, and you expect the diagonal to be more similar, less dissimilar than off-diagonal elements. And so that's a measure of the distinctiveness of the differences in the neural patterns. And you see that that is in frontal cortex stronger than for the experts and for the controls. And then we also did some correlations with behavior. And there again in frontal cortex, we see very clearly that the correlation with behavior is higher in the experts than in the controls. In higher visual cortex, whether you see it or not depends on where you put the statistical threshold, but it's clearly not so pronounced. So these three effects are most clearly there in frontal cortex. We also looked a little bit in neural networks, whether we can see similar changes like that. We used the most common architecture, the LXNet, and we looked what kind of representation we see at the end of this network when a network has not been trained, then when it has been trained on ImageNet, and then third, when there's further training happening with a small BERT database. And what we see basically is that the more training the network gets, the clearer, the more distinctive these representations become, as the clearer the diagonal becomes. And also you start seeing correlations with the human representations. Okay, so to summarize, expertise results in more robust neural representations. It comes for at least three changes: more consistency among individuals, more distinctive representations, and more correlations with behavior. And these effects are most clearly there in frontal cortex. Okay, this was a very quick run through. If something was not clear, if there are questions, then please feel free to ask them now.
Thank you very much, Hans. That was a really interesting talk. Let me see. We have a question here. Sorry, let me just bring up the... It's okay. We have a question here from Minya Zan. How big is the frontal region of interest? Do you think there are specific but smaller regions in the frontal cortex related to categorization? Mm-hmm.
Yeah, it's a good question. We used almost all of frontal cortex, like we also did in the earlier study, but we also looked in smaller regions, like, for example, dorsolateral prefrontal cortex. And it seems that this is a general effect that you see throughout several regions. Now we do not take all voxels, it's only the voxels that are active in the task that people were doing with the stimulus, so do a similarity weighting. So of course there are certain regions in frontal cortex that are not active at all. But for example, dorsolateral prefrontal cortex very specifically seems to show similar effects than when you take the whole big region together.
Great, and thank you. And we have another question here from Jules Brochard. Would you have an idea of how neural circuit may adapt with respect to expertise?
I'm not sure what Jules means with the neural circuit. Does he mean the brain regions that are involved, or does he mean at the level of the microcircuitry, like the columnar structure of a region and how excitatory and inhibitory cells connect to each other? I don't know exactly at which level he sees that. Of course, the neural circuitry at the most general level, which regions are involved, like I said in my introduction, that has been the subject of quite some investigation already. And studies have clearly shown that indeed the regions that are involved change when people acquire expertise. But which regions are getting more involved, that depends on the domain of expertise. Like I also mentioned, in at least some cases, you see that the fusiform phase area seems to be involved, but that's clearly not the case in all areas of expertise. So it really depends from the expertise area where you see the changes. The effects that I'm showing now, you could see them as being more general in the sense that when a region is then changed to expertise, I would expect that those representations would be changed in ways that are similar to what I've shown here, like more robust representations. And more robust can then be the consequence of at least three things, the three things that I talked about. But I would expect that this effect would not necessarily be always in the same region. So if you have another domain of expertise, other regions might be involved, and then you might see those effects in those regions. To show a very clear example, if it's an auditory level of expertise, like a musician listening to music, I would expect these kind of effects in auditory cortex rather than in high-level visual cortex, to the extent that we had effects in high-level visual cortex. So I think yeah, it will depend on that.
Yeah, we still have a few more questions. Here we have from Avital Hahami. Thanks for the talk. Why do you suppose there are group differences in the diagonals? Are the representations more distinct or less noisy in experts?
Yeah, why you can take that in very different levels, but I would say that, I mean, what it exactly shows us is that it makes a difference whether you repeat the same condition or where you look for difference between conditions. So the differences are more clearly represented if you have a clearer diagonal. So that's what the data show. Why you then see it? Well, I could rather just rephrase what I just said. I'm not claiming that these three effects that we see are totally independent of each other. To some degree, all three effects are somewhat also reflecting like noise or reliability of the data. And so you could say you just have more reliable representations. So I used the word more robust because reliable is a word that has some specific meaning, but yeah, they could also be somewhat related to each other, these three. And I would say in general, the representations are clearer. That's for sure.
Great. Thank you. So we have another question here from Haida. Do I understand it correctly that you start to see greater within category similarity in frontal cortex? Do you think that high-level visual cortex preserves the subordinate level differences between same category objects, and that this is the reason why you see these clear patterns mostly in frontal? Or did I perhaps misunderstand the data?
Yeah, I think that's a good question. I think so. We see greater reading category dissimilarity, and so that's important. It's a dissimilarity, so it's a difference that becomes greater and better represented in frontal cortex. And indeed, we see it less clearly in high-level visual cortex, but you see some trends. In particular, the greater consistency between experts is also there in high-level visual cortex, and greater correlation with behavior. Like I said, it's weaker than in frontal cortex, but I wouldn't dare to say that it's not there. So it's more a matter of degree than all or none, this difference between frontal cortex and high-level visual cortex. So it might also be a matter of us being able to measure the differences. But I also like the interpretation that Haida gives to it, and the solution or the hypothesis that she brings forward. I think it's an interesting one, and we could investigate that further. So thanks for the question.
Okay, I think we maybe have time for one more question from Ineke. At the end stage of training the neural network, there is a correlation with the human representation. Do you mean the representations from the frontal cortex, or also with the representations from the higher visual areas?
Yeah, I kept that a little bit fake because of time, but indeed, it depends on how you look at it. If you do correlations with the behavior of people, then you see that these correlations are getting better. Yeah, once a network has gotten training, group difference in terms of finding better correlations with expert data and with novice data, that is something we most clearly saw for the frontal cortex data. So the frontal cortex of experts correlates better with what you see with a network that has been trained on birds compared to a network that has not been trained on birds. And the correlation is also higher than the correlation with controls.
Great. Well, thank you very much, Hans, and thanks everyone for the wonderful questions. That was a great discussion. Okay. So our next speaker is Dr. Miguel Santa, who is also at the KU Leuven at the Department of Brain and Cognition at the Leuven Brain Institute. Miguel is a postdoc in the laboratory of Johan Wagemans, and today he's going to be talking about Object categorization and response bias flexibly change depending on cross-spatial scale coherence and task demands.
Okay, something else happening. My screen not sharing properly?
We can see your screen now. It's not at the slideshow though. It's your.
What now?
I think we have some kind of presenter view that we're seeing.
Yeah, it's the presenter view.
Okay. About now?
Yeah.
Perfect, yeah.
Cool. Okay, thank you for that introduction. And as Elaine said, I'll be talking about some aspects of object categorization. And how these things interact with cross-spatial coherence and task demands. So it's good that I follow Hans because he already talked about some things that I'm going to describe now. But basically, many studies have shown that categorization does not always necessarily follow a strict fit for the hierarchy. For example, like Hans showed, studies investigating expertise in certain visual domains consistently show that experts can perform categorizations of the highest level of specificity as fast as categorizations of the middle level of specificity. So you can think of how bird experts can name the species of a bird as fast as naming any given type of animal. One potential explanation for that could be that visual experts are able to pick up relevant visual features in the coarser scales and then predict where the finer scales are and then jumping ahead in the visual processing pipeline. So if we are able to disrupt these relationships between the coarser and the finer scales, we could have a window into how these configurable properties present in different spatial scales interact with task specificity and response certainty. So to do that, I use this image manipulation algorithm called the Eidolon factory, which basically decomposes an image into its scale space representation. If you have questions about that, we can talk about it later. But basically, it just creates this stack of blurred versions of the original image. And then in order to get a handle on the coarse and fine parts of the image, it basically just subtracts one layer from the other iteratively until you get a full stack that goes from blobs to fine edges. Then, in order to disrupt these scales, we actually introduce random disarray fields that deform the appearance of the image. This is called a parameter reach in this algorithm, and it's basically just a measure of pixel displacement. But the crucial part of this algorithm is that we can control how these arrays interact with each other. So take, for example, this cartoon example of an original image. The larger circles represent the coarser scales, and the smaller circles represent the finer scales. If we contain the deformations of the finer scales to the coarser scales, then we get an eidolon. This is what we call this images, where the edges are kept, but the appearance is deformed. However, if we then let the finer scales behave independently from each other and from the coarser scales, we get an incoherent eidolon, where the edges are diffused, but we still deform the appearance of the image. So in my task, I did that to images of animals, peoples, and objects. And I presented these images in a smooth sequence going from very disarrayed to original image over 15 seconds. Participants were allocated to two between subject groups. In the first group, they had to detect the presence of a human face among these two distractors, so a basic level categorization. In the second group, participants had to detect the presence of any type of face, so human and animal, among the distractors. For their task, they had to stop the sequence of this array at two levels of certainty. First, using their gut feeling to respond, where they would lift the spacebar and say yes or no, the target is present. And then by pressing down the spacebar again, they would be able to they were instructed then to stop only when they were sure about the appearance of the image. And here you can see how the information progressed over time in a logarithmic. So this is just an example of how it will look like. Hopefully you can see the images becoming clearer over time. Yeah, that's good. So now let's look at the stopping point results. So here we can see the results for the gut responses, where they stop. We find that a basic level advantage where human faces were detected earlier than any type of face. But we didn't find any other effects of our disarray. However, for sure responses, we actually see the opposite effect, where human faces actually became participants in the human face detection group actually took slightly a bit longer to be sure about their response than those in the any face condition. But we also find a small effect of the coherence of the disarray, where coherent sequences where the edges are kept actually impair the detection of targets. Then we also looked at signal detection theory measures, but only for the window is covering, but only for gut responses, because show responses were at ceiling of performance. So we looked at D prime, but we found no significant effects for this, as you can see from the plot. But we also then look at response bias, and the results are more interesting here. We found first that, okay, human faces, participants in human faces tend to have a larger tendency to say yes, that the target is present, but that's normal because if you say look for a face, they're likely to see faces everywhere. But the interesting result is at this level here, it seems that the presence of edges actually increased the tendency to say yes, but for the any face group, actually made participants behave unbiased. So they were actually performing at the mean of that distribution. And this effect here, or this increased tendency to say yes that is observed here, is larger for the any face group than for the human faces group. So how do we interpret these results? First, we propose that in line with previous research, the basic level is the most likely default entry point, but it could be that it depends on the mode of viewing that the observer is employing. Superordinate representations may be only triggered when sufficiently similar visual feature information accumulates so that you know that you are looking at a larger group of similar features. When thinking about the response bias results, we see that there is an interaction between coherence and group assignment, which highlights that tasks demands can affect the way that global and local visual information is being abstracted. And if you think about the responses at the gut level from the response bias as well, we propose that the visual system is rely on matching the local structure to appropriate templates to build a visual representation of the incoming stimulus, regardless if it's the right stimulus, the right target or not. The edges are important, is what's giving texture to your decisions in a more poetic way of explaining. So in conclusion, it seemed we offer a bit of a slightly different twist to the traditional view of these abstraction levels in the visual hierarchy, but we also seem to support the initial assertion that visual categorization does not necessarily follow a strictly feed-forward pipeline. And thank you, that's all I have to say.
Thank you very much, Miguel, for a great talk.
So
if any of our panelists have any questions as well, you're welcome to unmute yourselves. So maybe I'll just ask whether you could talk a bit more about what you think, why the response bias was so affected by the coherence manipulation, and what the.
Yeah, that's a good question. So we think that it's mostly because when the edges are present on the image, you get lots of categorizations get triggered, regardless if they are of the right category or not. So depending on the group, that seems to increase their tendency to be that the target is present in comparison to the incoherent condition. Because when the edges are deformed, you don't have distracting pieces of information that are aligning themselves to give the impression that something is there. So I can say that anecdotally, many participants reported back after their experiment that after seeing the coherent sequences, they thought that they were seeing a specific image, but then it turns out that it was completely different. I also didn't report something which is at the end of the sequence, participants were asked to rate how surprised they were by the appearance of the image. So the whole sequence, agnostic of the categorization, we just asked about the specific of the image, and we found that coherent sequences actually induced more surprised ratings. So it kind of supports that argument.
Very interesting.
I can show the results if you want for other people. And I have it in a hidden slide.
Yeah, well, if you want to.
I mean, if there aren't any questions, it's fine. Doing something weird. Hopefully stop. Not stopping. I don't know why, but I'll stop sharing because that's strobe attack to someone.
Okay. Okay, well, if there aren't any other questions, we can maybe move on to the next speaker. Thank you again, Miguel, that was a really interesting talk.
Yeah, really interesting. Very complicated looking, at least. I hope I've been moving around trying to look for Wi-Fi, and I hope I'm audible at least.
We can hear you.
Okay, that's good. That's good. That's something.
Okay, so our final speaker today is Tessle Blum, who is a PhD student in the lab of Dr. Hinze Hogendoorn at the Melbourne School of Psychological Sciences in the University of Melbourne. And she's going to discuss the time course of prediction formation and revision in human visual motion processing. Thanks.
Yeah, so thank you for the introduction. Yeah, so we're moving a little bit away, but we're sticking with the little bit of the classification analysis bit. So yeah, I'll be talking about how we form in the time course of prediction formation and how we overcome prediction violations in the temporal domain. And the reason that we are interested in this in our lab is that because of neural transmission delays, because the transmission of sensory information, so any sensory information, but we are mainly focused on visual information, through the visual system simply takes time. And that means that any available sensory information that we have available is lagging behind the present moment. And this wouldn't necessarily be an issue if we weren't living in a dynamic environment. So you can imagine if things are moving or you have to catch a ball, knowing where that ball is, accurately being able to localize it, the object is a necessity in order to interact with it. And what's been proposed is that the extrapolation of a position of a moving object, so having past information on the trajectory and extrapolating it into the present moment, can help overcome this. And by doing that, these predictive mechanisms can actually effectively decrease the processing time that are associated with the processing of these objects. And this has been shown in neurophysiology research, where in cats and macaques, where V1 just responds quicker to moving stimuli than to stationary stimuli. And it's also been shown in humans using EEG. So the question that this project tried to answer is how much information about a motion trajectory is required to see a latency advantage, and where along the visual hierarchy does this latency advantage emerge? So for which neural representation can we see this latency advantage? And this latency advantage, what I mean with that is that the visual system just responds quicker to a predictable stimulus than it does to an unpredictable stimulus. And to look at that, we had a very simple paradigm. So we just had this apparent motion sequence where we had this wedge. I don't know if this translates well over Zoom, but what you see is a wedge that moves around fixation. It has 8 positions. And because we present it every 100 milliseconds, you get this apparent motion illusion. And what we then did is that we collected EEG while participants looked at this stimulus move around, and we trained LDA classifiers. So we did multivariate pattern analysis, where we looked how can we train these classifiers to learn to distinguish the location of this stimulus. And we did that in a pairwise fashion. So you train position 1 versus 2, and you do that in all ways. And what we found that, yeah, indeed, our classifiers are able to learn the location of the stimulus. And what's important is that we train these classifiers only on the first stimulus in a sequence. So the fully unpredictable, it would start whenever, wherever. So it's an unpredictable presentation. And what we then asked is these neural representations that we've now trained it on. So every millisecond since stimulus onset, when are these representations activated when they're just driven, or when they're driven by predictable stimuli? So when the stimulus is further down in the sequence, so just the motion can predict the location of the stimulus. So we looked at the first 4. So we had stimuli going on and on and on, which ended up being a slight waste of time because we only cared about the first 4 where we saw differences. So we look at all of these individually and see when are these neural representations activated in these stimuli. And what we found is that on the x-axis here, you see training time points. So that's the point in time. So when we train the classifier, so what we can basically look at that as increasing in neural representation. So 70 milliseconds is the earliest time point where there's information, and then it progresses to later time points. And on the y-axis, you see the latency of the peak performance. So how well, at what point does the classifier reach 80% of its peak? We picked 80 because it's just less noisy than the absolute peak, but we've done it across all thresholds, and there's no meaningful difference in the results. So what we see is that for the earliest training time point, so the earliest representations, there's no difference in latency regardless of the sequence position. So regardless if it's the first or the second or the third one in the sequence. So predictability doesn't modulate the latency there. But then from about 95 to 100 milliseconds, we see that the third and fourth stimulus are starting to peak earlier than the first one. So they're represented earlier than an unpredictable stimulus. And then from about 125 onwards, we see that also the second stimulus, the stimulus in the second sequence position has a latency advantage. And that's especially an interesting one that we didn't expect, because the second one isn't predictable yet. The first one can appear anywhere, and then the sequence can go clockwise or counterclockwise. So the second one isn't predictable yet. Only from the third one onwards, you have enough information to predict the location of the stimulus. But what has been found, and what fits with this finding, is that there's such a thing as an omnidirectional within layer wave of sub-threshold activity. So you can imagine this first stimulus is presented, and then within the layer, it can trigger this sub-threshold wave of activity, reaching the receptive field of the second position. And then when the second position is presented, the time to peak is shorter. So you get this latency representation, this latency advantage for later representations. And then when the stimulus is fully predictable, so that's from sequence position 3 and onwards, we see this additional latency advantage that can be explained by descending prediction. So we see these two kind of predictive mechanisms interact. So we see differences across neural representations and across sequence positions. And to then look, are there also downsides to having these predictions? Is there a difference in latency when these predictions are violated? We also had the stimulus reverse. So we had it going for at least 2 cycles, and then at a certain point, it would reverse directions. And then you have, again, these 1, 2, 3, 4 sequence position after the reversal. And what we find again is that from about 100 onwards, the third and fourth stimulus have a latency advantage of the first, but now also the second stimulus has an immediate latency advantage, which can be explained by the fact that there's just more information for the second one, since the reversal, the final one before the reversal, can add information to this motion trajectory. I don't need to go into this if we're running out of time, but what we also compared is then if a stimulus is unpredictable, so at the start of a sequence, or if it's violating a prediction, so that's the first one after reversal. And we find that indeed a prediction violation takes longer. If there's a latency disadvantage for a prediction violation compared to an unpredictable stimulus, and that's in the neural representation that's affected by these descending predictions. Yeah, so that's what's shown here. So there's a latency advantage when a stimulus violates a prediction. So yeah, that I want to thank my 2 supervisors, Stefan and Hinze. And then if there's any questions, I'd happily answer them.
Great talk, Tessle. Thank you.
Thank you.
Okay. So I was wondering, is this decoding, is it done on the whole brain, or did you focus on visual areas?
No, we did the whole brain. Yeah. So we just put in all we did the 64 channels, so the 64 channel EEG and all 64 4 channels went in, and the external ones. Yeah, so it's whole brain. And then presumably the only information difference would be in the visual cortex, but that's an assumption, and never really know if that comes true or not.
Hans, did you have a question? Or?
Yeah, if there are no questions from the audience, in several plots, I was wondering, you summarize everything by looking at this, how do you call it, latency of the peak performance.
Mm-hmm.
I mean, there's only one way to characterize it, and I could imagine that there are certain aspects of the time course that you're losing or not characterizing by just the latency of the peak performance. I mean, is there anything that you might be missing there, or might you get a different picture if you look at something else, or is this the only thing you can do?
Well, the way we do it now is that we can look at, so we have this CAI, the peak performance. You can look at different time steps, right? So we look at 80% now, but the peak shows something different than if you look at 10%, for example, or 20, because you get the build-up. But they all showed fairly similar stuff. But what we also did with this paradigm in the past is that we looked at when we emit the stimulus at the end of a sequence, we can actually decode the anticipated location of it. So we actually also can look at the amplitude of the signal. But the amplitude differences are very small if the stimulus is actually presented. If the predictive mechanisms are negligible compared to the actual sensory signal. Yeah,
okay. Which by itself is also an interesting observation, I guess, right?
Yeah, so that was the first step that we did. So we've published that beginning of this year. And this then the next, looking at the temporal domain.
Okay, thanks a lot.
So I was wondering if you think that, or if you've looked into whether these predictions would have an influence on behavior, like if they.
So in the temporal domain, we haven't done that yet. But when I talked about the amplitude and how we kind of pre-activate this anticipated income, what we find, or what we're in the process of doing now, is looking at, okay, so can that pre-activation kind of bias perception when there's a very low sensory information stimulus, so either very low luminance, low contrast, can actually this pre-activation of these neural representations, of these sensory representations, bias perception in that way. But that's still in the works.
Sounds really interesting.
Yeah, I think so, yeah.
Just a really technical question. Just the decoding approach, the way you do the pairwise decoding, is that standard, or is that because of your specific interest in the timing comparison?
I think that's more, I don't think it's very common, but I think it's very determined by this paradigm. Yeah. Because if you look at the sensory representations of the one that's here and the one that's there and there are going to be super similar. So trying to actually decode those is going to be very difficult. But if you then decode them all in pairwise, you get you average or you summate, whatever, that doesn't change much. You actually get a much more robust measure than just trying to decode two that are very similar. But I don't think it's very standard.
Okay, I think we have a question. Is this paradigm somehow showing something related to mental imagery? No.
Would it be related to mental imagery? I'm not completely sure. What has been done with a similar paradigm is where they have had this. It also comes out of Melbourne, where they have this stimulus go around, and then they've stopped it, and they've ask people to keep imagining it continuing without the actual stimulus being on the screen, and they're also able to decode the imagined location of the stimulus. So it's very cool. What I'm not doing much with mental imagery, but yeah, it's a pretty powerful paradigm in that sense, because obviously when you're trying to decode in the visual cortex, position is such a strong difference between electrodes. So it's a pretty powerful paradigm in that sense.
Great.
Right.
Okay.
Thank you very much for that.
Thank you. Thanks very much to Tessle for a really, really interesting talk. And I guess we can finish up there. So I'll thank all our speakers again. I think that was a really good session.
Thank you.
Thanks, Elaine, for all the moderation.
Yeah, thank you very much.
Yes.
Thank you.
Thank you, Elaine.
Yeah, and the organization.
Just once.
Sure.
Yeah. I think
Perfect. Perfect. So I think we are right on time. And we can start. Hello, everyone. I'm Valentina, and together with the great help behind the scene of Faruzan, we are here to bring you 3 great speakers for this session. We have Lei Zhang from Vienna, Ankani Chattorai from Rochester, and Annalisa Gerd from Osnabrück. Each of them will have 5 minutes to present their work, followed by 10 minutes for your questions. So please write your question in the Q&A section at the bottom of your Zoom screen. And it will be my pleasure to read them to our speakers at the end of their presentation. But also don't forget to check the chat, because we will be sharing with you some link and some information on our speakers. So with this, I'm gonna give the stage to our first speaker, Lei Zhang, a postdoc at the University of Vienna under the supervision of Prof. Klaus Lamb. And he will be presenting us his work on how the left temporoparietal junction causally supports goal emulation in observational learning. So please, Lei, the stage is yours.
All right, great. Welcome, everyone. Thank you for joining. My name is Lei Zhang, currently working as a postdoc at the University of Vienna in Austria. And today I will be presenting the left temporoparietal junction causally supports goal emulation in observational learning. Because this is, in fact, a follow-up TMS brain stimulation study, I will actually first present some brief results from the main study and the rationale why we do the TMS, and then some preliminary results from the TMS dataset. So we focus on social observational learning, and observational learning is very important for our life, everyday life. Suppose if you go to one of the Chinese restaurants for the very first time, you have no idea how to use the chopsticks. Well, the best way to learn is to look around to see how the others are using that, and then try to practice, and then from some trial and error to improve your finger motor skills to use the chopsticks. And then the researchers often also use reward learning to study observational learning. So instead of doing some tasks by yourself, you could see how the others learn the association between a stimuli and the outcome to get a better understanding about the association. So this has been done in both humans and in animals. I won't go into the details. And there is also a quite recent review from Andreas Olsen's group, and they also highlights the brain regions that are related to observation learning, including one of the area, the temporoparietal junction, that will be the focus of today's short presentation. So how did we study observational learning as the main study? So we used a simple 2-armed bandit task. So there are 2 choice stimuli. So one of each of which is associated with a reward probability. So 2 symbols, you choose it, and then to see the feedback, reward or punishment, and then you learn from trial and error to work out which is better, which is worse in the long run. So here I show, what I show is the reward probability. You see here, it goes up and down. So in fact, this is a reversal learning. So what we ask participants to do in the lab is that in the first stage, we present the participants the 2 options. They have a choice on their own. And then afterwards, they could also see the choices from 4 other people. And then we ask the participants, do you want to change to the opposite, or do you want to stay with your previous choice? And then eventually they will also see the outcome from both their own choices and also the other co-players in the group. And we did a fMRI study. So they are real participants. Every time we ask 5 participants to come into the lab, 1 stay inside the scanner, and then the other 4 are sitting outside at the adjacent room. Okay, good. So we focus on primarily in this short presentation, the second stage of the task. So here, when after their own choice, when they see the choices from the others, what is happening in behavior, also in the brain. So not so surprisingly, we see that in this red curve, the more opposing choices the participants are seeing from the others, the more likely they are switching. And this is, I guess, quite straightforward to understand. And then we also look at the accuracy, both of the first choice and also the second choice. So we see that the accuracy on average of the second choice is higher than the accuracy of the first choice, meaning that after considering the social information, the either switch or not, but on average, the second one is better than the first. That also means considering this social information improves the learning. And then we also look at the same stage and then the brain response, and we actually found a map of brain activation. And here's only an illustration, not the exact map. So the key areas, including the bilateral TPJ, so temporal-parietal junctions. And what this means in terms of observational learning. So in the literature of observational learning, there are usually 2 strategies. One is called choice imitation, and then the other one is called goal emulation. And we were thinking that this result is reflecting goal emulation, because if they do choice imitation, they might just simply copy the most frequently selected option from the others, simply copy their choice without thinking too much. But if they are trying to infer why they are doing this, to infer their mental states, and then this is like classified or categorized as goal emulation. So to long story short, we did a study, we found bilateral TPJ, and we were asking, is this something causal? How can we establish some causal relationship between the brain activation and the behavior? So again, we used the similar paradigm. We used the within participant double-blind design, and we stimulated 3 brain sites. So one is the left, and then the other one is the right TPJ, and then there's also a control in the middle of the brain. Same task, same analysis. So here we see that here across the 3 stimulation sites, what we analyze in the first place is the choice switch probability, the one I showed. So previously, the more opposing you see, the more likely you switch. And then we see, especially here in this one, so in the left TPJ condition, this switch is less relative to the control and also relative to the right TPJ. And then the second, we analyze the reaction time. So same screen here after they see the choices of the others, how fast or how slow I respond when deciding my second choice. Either stay or switch, I don't really care in this analysis. I just analyze the reaction time. And you'll see quite interestingly, the right TPJ and vertex control, vertex control condition, they are pretty much the same, the pattern. And for the left TPJ, they are slower. So meaning that the more opposing choice you see, the more slower your responding is, not faster relative to the other 2 conditions. And then there is a third analysis that we did, is that we look at the accuracy after switching or staying, especially switching. So the first analysis I showed you that when they see opposing choices, they do not switch that much. But what if they switch? How will the accuracy look like? So in these occasional switch cases, their accuracy is actually decreased, especially if you look at here, this line relative to the RTPJ, there is an interaction effect. So it's pretty interesting. And we also use the computational modeling to quantify what might be the cognitive processes of the this learning task. And the detail I also will not show here. There is a full paper about this, and I can share it also on the link. So what we focus on is this stage, how the instantaneous social information affects the choice of the second choice. So what we did importantly for this short presentation is that we are trying to link the association between the model parameter and the behavior. And you'll see here that the strength between the model parameter and the behavior, especially here, this one is the weakest relative to the control and also for relative to the RTPJ. So to conclude, and also some discussion points. So we used a simple TMS study to have a follow-up of our previous main study, and disrupting the activity in the left TPJ resulted in the reduced choice accuracy switch probability, reduced the choice switch probability first, second, slower response speed, and third, reduced choice accuracy when individuals were contradicted by the choices of the others. And then the computational modeling further reveals some learning process and its link to the behaviors. And some discussion points. Well, so we found the left TPJ, to keep a long story short. But most studies, if you know the literature of social neuroscience, they report mostly the right TPJ, or at least the bilateral. And we were wondering about the association, the connection to the literature. And the second, I asked some brain stimulation experts, and they say, well, if you stimulate one side, it never stimulates only that area. It stimulates a network. There might be a crosstalk. Maybe this is too much, but I don't know. And the third, we only did TMS, not fMRI. So the future work can also combine TMS and the fMRI to see some brain connectivities, for example. Thank you so much. That's it from my side.
Thank you, Lei. Thank you for the great presentation. Really a lot of data. You did a great job in trying to fit in the 5 minutes. That was a hard task.
I forgot to thank my collaborators and supervisors.
Of course, we know it always takes a village to do this kind of study. We know that. So we are open for questions. We still have, we ran a bit long, but we still have 5 minutes for questions. So please just, everyone should feel free to start typing in the Q&A. Maybe I can start, and since it was actually one of the points in your discussion, in my ignorance, I was expecting the right TPJ indeed to have more of an impact. So you mentioned that it might be a matter of your stimulation and this crosstalk. Maybe you want to elaborate a bit on that. Like, how could you go about teasing that apart? Like, what could you do to avoid that confound?
Not really sure, honestly. So we did a within participant study. This is one of actually the effort to minimize the between participant variability. And you could wonder that if there are data in the simplified word, let's say there are two sources of variability. One is the interpersonal variability between different participants. The other one is the within person, right? And then the people usually argue that the within is the between participants variability or variation is actually larger than the within. So that's actually one of the reasoning why we designed a within participant study design. If, on the other hand, if supposedly in the imaginary world we designed a between participants design, then it might be even noisier, I would guess.
Right.
Yeah.
Yeah, that's a good point. Do you know of any study with patients that have lesions either on the left or the right TPJ, and whether they would show this effect or not? Is that something that's been studied?
Yeah, that's also a question, I think. So at least from my field of interest, so social decision-making, social learning in particular, most of the brain stimulation studies, they had a right TPJ stimulation site and they control, either sham or vortex, doesn't matter. So I rarely see people stimulate also the left. Yeah, that might also be the reason. I don't know. I really have no idea.
True. So if any of the other speakers wants to jump in or has a question, don't hesitate, just motion me. Yeah, please, Hankani, did you? Yeah.
No, I think great work. I don't have any question at this point.
Thanks.
Yeah. And do you know what you're going to do next, Lei? The fMRI will be the next step?
Yeah, perhaps. Yeah, this can be done. I didn't mention the details. So we use the specific brain stimulation sequence that we used is called a theta burst, continuous theta burst stimulation. So it's not really online brain stimulation. So it works in this way. The participants come into the lab, we stimulate the brain for even less than 1 minute. And then the effect is expected to last for about half an hour. So usually what people do in this kind of protocol is that it actually overcomes some of the challenges to combine TMS and fMRI, right? So they come and then stimulate for within even 1 minute, and then they could go inside the scanner to see the changes, especially the connectivity patterns, functional connectivity patterns. Yeah. To
And
see
again,
how the TPJ is associated with other areas. For example, VMPFC, striatum that are supposedly to be related to valuation, learning, prediction error.
Right. And again, a question probably very naive, but you mentioned that the 5 participants, they're all like volunteers. 1 gets in the scanner and 4 are outside. And I'm guessing they don't know each other, right? So it's like social learning from someone you don't know. Would you expect any difference if they actually are familiar with if they're your friends or your family? Do you think you would be more affected?
Yeah. We will guess so. Yeah. One of the participants recruitment inclusion criteria is they say you don't know each other, and if you do see as a group, then better sign up different sessions. So we try to avoid this familiarity effect. But it might be interesting to do another study, and one group is like familiar people, family members, friends, and another one purely strangers, and then to see the comparisons. And the potential analysis can also be to use representational similarity analysis, and also so there is kind of a trend to use one brain activation to predict another person's brain activation. So intrapersonal analysis, that can be very interesting. Yeah.
That is incredibly interesting. It sounds like you have experiments for the next 10 years to do, and that we should all be following you. We shared your Twitter account, and we will be following your publication because definitely there's a lot coming out from you. Thank you very much for your presentation.
Very glad. Thank you.
We can now move to our second speaker for this session. Ankani Chatorai is a graduate student at the University of Rochester working on the implication of approximate inference in visual perception. And she will be presenting us her latest work titled A Confirmation Bias on How Human Actively Sample Sensory Information. Thank you very much, Ankani.
Okay, so I will start. Okay. Hello, everyone. A huge thanks to all the people organizing and part of this wonderful conference, and thank you for letting me present this work. So today I'll talk about confirmation bias in how humans actively sample sensory information. This is joint work with Sabyasachi Shivkumar, Ra-Yong Su, Catherine Moon, and Ralph Hickman. Okay. So first, let me briefly introduce the question we are addressing here. Over the years, several studies investigating temporal integration of evidence have shown that human subjects often are biased to weigh early evidence more into their decision. Now, we explained this in a recent work via approximate inference in a hierarchical model, and the approximate nature of inference in our model initiates a feedback loop between the decision area and the sensory representations, making us see what we expect to see. So now we try to extend this and understand if humans also actively seek information confirming their beliefs. Our project builds on recent work by Yang et al., where the authors have already shown that subjects do indeed make eye movements in a goal-driven manner in discrimination tasks. And we extend this and design a paradigm where subjects incorporate information from the periphery to determine their Sakat targets. Okay? So let me now elaborate the 2 tasks that we examined here. The first is a gaze contingent task. In this task, the subject first fixates on a bandpass Gabor stimulus, either oriented plus 45 degrees or minus 45 degrees, shown with a blue cross. While subjects fixate, two more stimuli appear in the periphery. Now, after 250 milliseconds, the subject is allowed to saccade to one of the peripheral targets. After the saccade lands in one of the chosen peripheral stimulus, the non-saccaded stimulus and the already fixated stimulus disappear, and two new peripheral stimuli appear. And the subject again makes a saccade, and this continues for 3 saccades, after which the subject has to report the dominant orientation in the trial. Okay? The second experiment is very similar, but close to a more natural scenario. The stimulus here is stationary and consists of 18 small ellipses, either horizontal or vertical, with an aspect ratio of 0.55. Subjects freely move their eyes for 1.5 seconds on the screen. After that, they report the most frequent category of the image. Okay. So now first look at results on human data for the gaze contingent task. In this plot, each yellow bar represents one subject, and in the y-axis, we plot the percentage of correct responses. Almost all subjects have close to 80% performance, confirming that they can do the task. In further analysis, for each trial, on any ith saccade, we compute the evidence accumulated by the subject from the saccades already made. This gives us an estimate of the subject's current belief about the correct category of the trial. Now we then check if the next I plus 1th saccade was made to a peripheral stimulus that had evidence confirming the subject's current belief about the correct category of the trial. Okay? So we call this the confirmation bias in evidence selection. This then allows us to compute the probability with which each subject saccades to peripheral evidence in favor of accumulated evidence so far. That probability is now what I'm showing you on the y-axis of the plot on the right-hand side. And each yellow bar is again one subject. And we find that almost all bars have a mean above 0.5. And this suggests that for human subjects, the integrated information from already fixated stimuli drives eye movements towards confirmatory evidence in the periphery. Let us now look at results on human data for the free viewing task. We first look at the psychometric curves for subjects, which confirm that the subjects could perform the task. We again computed the probability of saccading to confirmatory stimuli in the periphery for each subject in this free viewing task, which is shown in the y-axis, and each yellow bar is again one subject. And in this task also, we found evidence that subjects were systematically biased to make saccades to ellipses whose orientation agreed with their accumulated evidence at that point in time, because all the bars are above 0.5, as you can see. Okay. To model the selection of saccade location, we follow Young et al. For this task, an ideal Bayesian observer would saccade to locations that maximize information with respect to the trial category by computing a Bayesian active sensing score, which we abbreviate here as BAS score. Now, for every fixated location, the Bayesian ideal observer would compute this BAS score for each of the 2 peripheral locations and choose to saccade to the location with the greatest score. In this work, we hypothesize that the brain cannot compute the score exactly, but approximates it using sampling. Now, mathematically, the BAS score is nothing but the mutual information between category of the trial and category at the peripheral location. And it can be broken down into these 2 components. And we use sampling to approximate each of the 2 components. Okay? Now, the degree of sampling-based approximation of each of these 2 components will determine the type and amount of bias in saccade selection. Since component 1 is the total entropy, we hypothesize that this local information computation is presumably carried out in lower cortical areas such as B1, which has been previously modeled as a high throughput buffer with fine representation of local features. So this would then be analogous to drawing more samples in our approximation of component 1. However, component 2 is the noise entropy, which involves inferring the category of the trial and hence requires more global information. So we assume that this computation is done in higher cortical areas of the brain, where the representation is coarser. This would then be analogous to drawing fewer samples in approximation of component 2. Okay, with these assumptions, here we show some model simulation results. We generate subjects with different degrees of approximation of component 2, from blue to red to green. The approximation of component 2 becomes stronger. For these model-generated subjects, we again compute the probability of making bias occurs to peripheral stimuli in favor of accumulated evidence, shown in the y-axis. And we find that the bias was strongest in observer B, shown in green, who used least number of samples to approximate component 2. Whereas the bias was least or almost absent in an ideal observer I, who uses the most samples to approximate component 2 in this case. When comparing to human data, we see that the approximate inference model shows biases that qualitatively match the empirical findings from human subjects. To conclude, we investigate whether eye movements are biased when sampling new information in visual perceptual decision making. We test this in 2 tasks, a gaze contingent task and a free viewing task. And in both experiments, we do find some evidence that integrated information from already fixated locations drive eye movements towards confirmatory stimulus. And finally, the pattern of subject responses can be explained by an approximate inference model of information exploration. Thank you for your time and attention.
Thank you very much. Thank you very much, Ankani. Very clear, even for me that I'm not an expert in the field. Thank you very much. I was struck by the variability that you still see across participants.
Yes.
So I wonder whether you can elaborate on that, whether you are planning on getting more subjects.
Yes. So this is very preliminary work. We haven't gotten a chance to get more subjects yet. And the second thing is, as I described, our model shows that the degree of approximation of component 2 drives the degree of the bias. So, we haven't done model fitting yet on the human data. I just said it qualitatively. We want to first finish collecting all the data and probably then get the data to humans. And yes, so. We expect that that will show up as the number of samples being used to approximate component 2 in each of these subjects. So if someone is more ideal and uses more samples, we would probably see that they are not that biased, whereas others would.
Do you have any neuropsychological measure that you're planning on collecting on the participants and try to correlate with how much they rely on?
We are this is a purely psychophysical experiment and study that we had in mind. Yes, I mean, we haven't thought about it yet, but definitely our previous work on perceptual confirmation bias, we have a follow-up project on monkeys, where we try to cool areas to see if we stop feedback, what happens. So yeah, once we probably get done with the psychophysical part of the study, we might try to do something with human fMRI, but I'm not sure. Yeah, we haven't thought about it yet.
Right. No, it just makes sense because as you put forward, there's an hypothesis on which brain area might be driving which.
Yes.
So it would be extremely interesting to follow up with that. I'm checking the Q&A. Sorry, I don't want to get distracted and monopolize. And also the other speakers, or if anyone wants to jump on screen, I believe we have a couple of minutes if anyone wants to. Otherwise, I'm going to keep with my very naive questions. Okay. We have a question from Richard Lange. He's asking, do more biased subjects perform worse in the task?
We do. Yeah. It did not. We did not try to. Not really. We did not find a significant correlation between more biased meaning worse performance. Also because we have 0.7. 7 of the frames, 70% of frames matching the correct category. So there's a high chance that the first frame you see also is the correct answer for the trial. So yes, no, we did not find any
Right, accuracy was pretty high, right? Overall,
yes. It was almost, very, very, very close to 80%.
So are you planning to try anything harder, like any task?
The task itself is really hard because like we do not allow them to blink because we want to make sure they're using the peripheral information while making a choice, and not just blinking and not seeing what's there in making a conscious decision. So it's already a hard task, and we did some manipulations to see what was a regime where most people could perform the task reasonably well. And for now, we have converged there, but we might decide to change and make it harder.
Very, very interesting. And well, you said it's preliminary, but it's already very interesting. So good luck for the follow-up and collecting more data. And it's time for us to switch to our third and final speaker, Annalisa Gert. Anna is a PhD student in the Neurobiophysiology Lab at the Institute of Cognitive Science at Osnabrück, under the supervision of Peter Koenig. And her talk is titled Face Processing in Ecologically Valid Scenes, Characterizing Fixation-Related ERPs Under Free Viewing Conditions. Please, Anna, take it away. You're muted, sorry.
Can you unmute me?
Yes. We heard you for a second. Yeah, I think now it's working.
Thank you very much. Thank you very much. So what I will be presenting to you today is our results from a bigger project. So our goal ultimately is to check how well lab-based results in EEG experiments translate to the real world, because we always make these statements about the brain, how it works, but what happens when people are really acting in their environment. And we use face perception and the N170 as an example for more general experiments. So what we normally do in EEG experiments here are lab-based conditions. People are sitting in the dark. They are not allowed to move. They have to stare at the screen, and they are passively presented with, for example, faces. And their task is to just perceive the stimulus as what it is, for example, as a face. In our next step that we are doing here, the very same subjects are sitting in the lab, but they are allowed to move their eyes. So they are actively sampling their environment. We are presenting them with scenes of a local shopping mall, and their task is to just freely explore it. And then in the last step, our ultimate goal is we've got, again, the same subjects, and they are wearing a mobile EEG, they are wearing mobile eye tracking, all the recording equipment is in the backpack, and they are doing a free viewing experiment in that same shopping mall. So they are allowed to go wherever they want, they're allowed to look at whatever they want, and there we've got basically the full natural setup. Today I'm going to present the first two conditions, so a passive and a free viewing lab-based experiment. And then our classic passive perception experience, the participants are presented with either faces or objects for 300 milliseconds at the middle of the screen. And in our free viewing experiment, they are presented with scenes that are taken in the shopping mall. They are magnified so that the size, for example, of a face will be the same size as if they were in the shopping mall. And they are presented for 6 seconds. So we gather a lot of data, we gather a lot of fixations. And what we then do is, this is an example trial. So they start out in the middle here. They start out in the middle, and then they will look at a face first. And from there, they might go to another face, to the background, and so on and so forth. And what we're interested in is to look at the ERPs and basically a 2x2 design. So we categorize them by what they're currently landing on. Are they now looking at a face or not? And then also where they come from. So were they previously looking at a face, yes or no? And that all the data that we gather goes into a model. We either model the fixation related ERP, so the FERP. We've got the factor currently on a face, yes or no, previously on a face, yes or no. Then we've got a special case within phase, where people are making 2 consecutive fixations within the same phase. And then we also correct for the X and the Y position of a fixation, but also the saccade amplitude, because we know that these factors will influence our ERP very drastically. And we don't want that because there might be systematic differences between our 4 conditions. And importantly, we also have here this stimulus-driven ERP. So that is what you would normally do in a lab-based experiment. Stimulus comes on, what happens in the brain. And with our lab-based classic passive perception experiment, we can replicate previous results. So here, this is the N170, and it's located in the parietal occipital electrodes. And then when we look at the free viewing data, again here, the same electrodes, we again find a stronger peak when people are looking at a face versus when they're looking at a background. So we can replicate the findings that have been reported for over the last 30 years. Interestingly, these 2 results correlate. And what's also interesting is that the variability across, but also within subjects, is smaller when we have a free viewing paradigm than when we've got a passive perception experiment. What we now additionally want to do is we do not only want to look at this classic 4 electrodes, certain time range analysis, but we also want to look at all electrodes and all time points. And what we find is that when you currently look at a phase, your ERP will be much stronger. So this is in a lot of electrodes during the whole P100 and 170 complex. If we split by what was previously looked at, we find that when you were looking at a phase, you will have a diminished signal, so a more positive signal, even before fixation onset. So this is not an adaptation effect because it's not dependent on what I'm looking at now, but this is basically, did I see a phase before, yes or no? Then if we look at the interaction term, so people are looking from one phase to another phase, I find a weaker N170. When I'm making fixations and saccades within the same phase, I see again before 0, there is a weaker response. And this might be an adaptation effect because I have already been looking at this very phase before. To sum up, normally we have classic passive perception, people are sitting still in the laboratory. Then we can now replicate these findings with FERP, so fixation locked responses, not only stimulus locked responses that are much broader than previously reported. And what we also demonstrate is that the response will depend on the fixation history. So what did I look at before, which was not able, we were not able to show that in lab-based experiments. And then also the wild data of people running around in the shopping mall is also currently being analyzed. Thank you very much.
Thank you, Anna. Extremely interesting, and I see a lot of questions already coming up, and it's just cool new methods that, of course, we need more clarification, we need more details. So the first question is from Ilya Nundu, and concerning the free viewing experiment. Just to clarify the method, so you define the stimulus onset as when it appears in the scene or when the participant fixate on it and then start processing.
So the stimulus-driven ERP is basically the stimulus comes on and I'm triggering to that. And then everything else, the whole 2x2 design is based on when a fixation is made on that phase. And that is purely dependent on the participant. But of course, for the very first fixation, it might land on a phase, the stimulus driven ERP might be on a phase, yes or no, depending on how the stimulus looks, but that we also correct for.
We also have a question on the adaptation effect that you see for faces. Andrei Nikolaev is asking whether you see this adaptation also for objects fixations and not only faces.
So I assume you're talking about this one, the within phase effect, because this one is not adaptation because it's not dependent on what I'm looking at right now. So this one at point 0, I might be looking at a phase or at an object. while this one is purely dependent. I'm coming from a face and I'm looking at the same face. And so we know that this is true for faces. Whether we've got the same effect for objects, we don't know because we do not categorize them. So if you look at the scenes, we have bounding boxes for the faces, which were hand tagged because it's a very tedious task. And we did not, for example, mark every jacket and door and so on.
I see. I only have a bit of experience with MEG, and I'm constantly worried of movements and all these possible confounds. And I ask my subject to stay still. So I'm just trying to wrap my head around what happens when you have free viewing, or even more so, the third condition you only briefly mentioned, people walking around. How do you deal with the noise? And
oh God, yeah, yes, very good question. Can I present from here again, maybe? So, okay, you can see, so we've got our subjects, cap, eye tracker, but then for because we know steps will influence the signal, because of course everything shifts, right? We've got these pressure sensitive insoles that we put into the shoe of our subjects, so there we know when they made steps, so we can correct for that. We develop them ourselves, and they get like a continuous signal, and then we can use that to, for example, subtract independent components from that. And besides that, we're very lucky. So we're using the ANT system, and that is quite stable for example, cable sway. The backpack weighs 12 kilos, and we have regular brakes. We've got some more muscle artifacts, of course, but the signal looks suspiciously good. Yeah, it's very nice. I was so super surprised when I checked out the data. I was very worried, but we only have one subject that looks really bad.
Okay, that's really great to know. And are you focusing mostly on face perception, or now you are extending your interest, as we were saying, of objects and categories of objects?
I'm mostly interested in face perception right now, because we know this N170, and it's a very clearly defined marker that we can very nicely try to replicate.
Yeah, I agree with what Ilya is writing in the chat, that this is very encouraging for the future of cognitive neuroscience, the idea that we can go in the wild and record brain signal while people are actually doing something.
Yeah. I mean, the development of the setup and getting everything to work took 1.5 years. So it was a lot of work. But I assume with the systems getting better, it might be much easier.
Right. I don't know if the other speakers want to come back on and have some comments. I definitely learned a lot this morning from your 3 talks, and it's really interesting also to see how they fit together, right? To go from the carefully looking at as Uncanny is doing, at the biases we have with one little saccade, to go to free viewing in the world. Extremely interesting. Well, let me thank you very much for your beautiful presentations, and our attendees for joining. Everyone is clapping and sending virtual congratulations on the chat and online. There will be now a short break, and then Neuromatch will continue with the next session. But thank you all very much. Bye. Great job, guys. Bye.
Thanks for your talks, and we have to move on at 15 pretty promptly.
Just to be sure, can attendees come to the stage and talk or not? So for their questions, for the question and answer.
I didn't catch your question.
Yeah, for the question and answer part, I'm going to send a text to the attendees to make sure. Oh, now I hear my.
Hi, Christoph.
Hi, how are you?
How are you? I'm so sorry. Could you tell me how to pronounce your second name.
It's vitamin. It's easier
Vitamin.
for Americans because they always term it like vitamin C, but in Ireland and the UK that doesn't really work. So
thank you.
But my PhD advisor still gets it wrong. So it's he's never learned it over all the years. So.
Didn't even realize he'd asked the question.
Oh, okay.
Right, but now it's better.
It's better now, yeah.
So I'm going to stop sharing my video until it's my time to speak. I think that.
We're now live.
Okay, welcome everyone. Welcome to this session of NeuroMatch 3. My name is Elaine and I'll be introducing the speakers today. So just to first quickly remind you, if you have any thoughts or comments, feel free to share on Twitter with the hashtag NMC3. We have a fantastic lineup of speakers today. So first we're going to learn about reward seeking, and then followed by two very different talks about memory. The talks, so please use the chat to send messages during the talks, and also use the Q&A tool to ask questions of the panelists. And you can also upvote the questions that you're interested in. So please don't be shy. And so I'll introduce now our first speaker today is Elizabeth Hansen. Elizabeth is a postdoctoral associate working in the lab of Benarenkiel at the Baylor College of Medicine. And she's new to the area of behavioral state regulation, and so she'd be really interested in hearing your feedback. Okay, go ahead.
Okay, I'll share my screen. Okay, is that good? Thumbs up from someone? Okay. So, hey, like Elaine just said, my name is Liz, and I'm going to tell you about work I've been doing as a postdoc in Benarenkiel's lab, Baylor College of Medicine. And in this project, I use a combination of fluorescent sensors, fiber photometry, and behavioral state manipulations to look at cholinergic signaling within the basal forebrain during olfactory guided behavior. So I want to start by acknowledging the lab and especially my mentor, Benarenkiel. And I also want to highlight a super talented postbac, Katie Brandel-Enkrip, who's helped a lot with this project. So I mentioned we're studying the basal forebrain, but that's actually a complex of nuclei, which each include a lot of cholinergic neurons, sending long-range projections to different brain regions. And these neurons mediate top-down control related to attention and arousal. They're important for cognition, learning, and memory. And their degeneration is associated with both cognitive and sensory deficits in neurodegenerative disease. So I'm going to focus on a region of the basal forebrain called the horizontal limb of the diagonal band of Broca, or HDB. And the HDB is especially interesting because it sends both cholinergic and non-cholinergic projections to the olfactory bulb and to multiple cortical regions. So it has a lot of potential to control both first order and higher order sensory processing. And by understanding this basal forebrain circuit and how it controls olfactory processing, we can begin to understand the role it plays in cognition and how its dysfunction contributes to cognitive decline. So we mostly tend to focus on cholinergic projections and their impacts on downstream brain regions, but there's also distinct roles for cholinergic signaling locally within the basal forebrain. So this is a diagram of some interactions between cell types in the basal forebrain. And I just want to focus on this interaction between the cholinergic and parvalbumin GABAergic neurons. So both of these populations in the HDB send long-range projections to the cortex and the olfactory bulb. But we don't really understand how the parallel projection streams work together to regulate downstream circuit function. We do have some specific clues about how this circuit might be working from another subregion of the basal forebrain, the medial septum. And the medial septum actually has a similar circuit motif of parallel cholinergic and GABAergic outputs, but targeting the hippocampus instead of the cortex and the olfactory bulb. And the upshot is that together, the cholinergic and GABAergic projections coordinate this oscillatory activity in the hippocampus. And at the same time, they set the excitability to a level where you get meaningful information nested within the oscillations. And so the key here is that cholinergic and GABAergic output is coordinated by this cholinergic signaling within the basal forebrain, which directly activates the GABAergic projection neurons. So from this, we hypothesized that GABAergic output to the cortex and to the olfactory bulb would be controlled by local cholinergic signaling within the HDB. And in this case, HDB cholinergic signaling would be a critical component of olfactory information processing, potentially even underlying coordinated sensory and cognitive deficits in disease. So this led us to ask the question, how does cholinergic signaling within the basal forebrain influence complex olfactory processing? And so we've approached this in two parts. First, by asking what actually is the pattern of cholinergic signaling in the HDB? And then what is the impact of HDB cholinergic signaling on GABAergic output? So to address this first question, historically it's been pretty difficult to assay acetylcholine in vivo and in real time. So we took advantage of this new GPCR-based cholinergic sensor from Yulong Li's lab called GACH. And this allows us to directly measure changes in extracellular acetylcholine. So we injected a virus expressing GACH pan-neuronally into the HDB. And at the same time, we implanted a fiber optic over the HDB to measure cholinergic tone using a fiber photometry approach. And this allows us to directly assay patterns of cholinergic signaling in the basal forebrain in real time during freely moving behavior. So to get at the question of how these signals are tied to olfactory perception and decision-making, we imaged cholinergic signaling during the performance of an olfactory-based go-no-go discrimination task. And so the way this task works is that water-restricted mice are placed in a box with an odor delivery and a water reward port. And entry into both ports is monitored with an IR beam. And so mice are trained to self-initiate trials by poking their nose into the odor port, where they get puffed with one of two odors, S plus or S minus. And the S plus odor means that they can go to the reward port to get a water reward. The S minus means they need to reinitiate the trial. So once they're trained on the mechanics of the task, they can typically learn new odor reward associations within a single session of like 200 to 300 trials. So this flowchart just shows the possible trial progressions from odor presentation to the different potential outcomes. And as a mouse completes a trial, we can use the beam breaks at the odor and reward ports to determine when a mouse initiates a trial and when it seeks a reward. So using our knowledge of the trial outcomes and the timings of the port entries, we can infer the timing of certain behaviors and behavioral states within the task. So for example, trial initiation involves approach, active sensing, and odor perception. Then following odor presentation, there's a decision made and an action taken, either to seek a reward or reinitiate a trial. And each action comes with some expectation regarding the outcome. And finally, in at least reward-seeking trials, there's quantifiable feedback where they either get a water reward or they get a timeout punishment. So by combining this behavioral task with fiber photometry, we can look at patterns of cholinergic signaling in the HDB that correspond to these behaviors and behavioral state features within the task. So this is what the data looks like. These are individual trials from a single session aligned to the trial initiation times. And this is only showing the hit trials right now, but we can see a pretty clear pattern. So averaging across trials shows that as the mouse approaches the odor port to initiate a trial, there's a relatively flat baseline. But after the S plus odor presentation, the mouse is actively moving to the reward port. There's a sharp increase in HDB acetylcholine. Then once the reward is obtained, acetylcholine decreases again, and then actually falls below baseline some seconds later. So this turns out to be a common pattern across animals and sessions in the HIT trials. And when you compare trial types, you see similar and different patterns emerge. So here, the trials are separated by outcome, notably in the false alarm trials where the mouse seeks a reward incorrectly. There's a similar increase in HDB acetylcholine between the odor presentation and the reward port entry. And so that's shown in these traces in the blue trace, that's the false alarms. So to quantify this across animals, I used the slope of the signal after the odor presentation. And the slopes of the hits in the false alarm trials are reliably higher than the correct rejects and misses, implying that this increase in cholinergic tone is related to the reward-seeking behavior. Another notable difference here between the trial types is this suppression, which is only present in the hit trials. So to quantify this, I took the total area under the curve for the suppression below the baseline. And we found that the post-reward suppression is reliably larger in hit trials, which indicates that the reward delivery is required for the suppression, not just reward seeking or reward port entry. So what I've shown so far with this data is that we can use fiber photometry and the GACH cholinergic sensor to directly record real-time changes in acetylcholine and the HDB during behavior. And doing this reveals that cholinergic signaling increases during reward seeking, which you can see in both the hits and the false alarms, and cholinergic tone is suppressed following reward delivery. And I'm just going to touch on the second part of this question briefly, which is about responses in the HDB GABAergic output neurons. So to begin to examine this, we used a similar fiber photometry approach, but instead of GACH, this time we're imaging GCAMP specifically in the HDB GABAergic neurons. And so in recording GCAMP fluorescence during the same go-no-go behavioral paradigm, we observed a similar bidirectional change in HDB GABAergic neuronal activity. However, this pattern is subtly different compared to the changes in acetylcholine. So, for example, the increase in GABAergic activity after trial initiation is more tightly linked to the odor presentation itself. So in 3 of the 4 trial types, there's a relatively sharp response to the odor, and this is true for S+ and S- odors here. And where the response gets muddied is in the miss trials. So on average, the misses have much more variable and significantly longer times to peak. And so what we think this suggests is that in the miss trials, the odor wasn't appropriately detected or perceived. And this is directly in comparison to the false alarm type of errors, which require that an odor be perceived or detected but incorrectly identified. So these data imply that activation of HDB GABAergic neurons is tied to odor detection instead of reward seeking specifically. But then mirroring the change in cholinergic tone, we also find that GABAergic neuronal activity is suppressed following reward delivery in the hit trials. So that you can see in the green trace here. And that's quantified here. So to end on, I wanted to come back to the main question and to this framework for understanding behaviors in the go-no-go task. And I want to argue that by directly measuring HDB cholinergic signaling during behavior, we can begin to understand the role that it plays in olfaction and olfactory guided decision-making. So far, we've shown that acetylcholine ramps up in the HDB between odor presentation and reward port entry. And because this happens in both hits and false alarm trials, it indicates that cholinergic signaling follows reward-seeking behavior. And it may also reflect an internal state of expectation. So if this is the case, increased cholinergic signaling may play a role priming neurons in the HDB to respond to anticipated feedback signals, for example. And at the same time, we find that reward delivery transiently suppresses HDB cholinergic tone, suggesting that the signal is related to positive reinforcement. And intriguingly, the suppression below baseline also implies the presence of this baseline cholinergic tone, which can be bidirectionally modulated. And then when we look at GABAergic neuronal activity, on the other hand, we see a more general response to odors. And we think this is specific to active sensing because we don't see that kind of response with passive odor presentation. So the animal has to be like actively trying to sample the odor. But we also find that GABAergic neuronal activity transiently decreases after reward delivery, similar to what we found for HDB cholinergic tone. So we hypothesized at the outset that local cholinergic signaling controls HDB GABAergic projection neurons. And these data would suggest that cholinergic tone and GABAergic signaling are correlated, but only under specific circumstances during specific phases of the behavior. And the transient suppression of each signal seems to be involved in the positive reinforcement. And so I think this is surprising and pretty interesting because it implies some level of tonic activity in HDB GABAergic neurons, which is directly tied to changes in local cholinergic tone. And so we're working on teasing this apart now and looking at like GABAergic subtype specificity, projection specificity, that sort of thing. But that's where we're at now.
Excellent talk, Elizabeth. Thank you.
Thank you.
So we have a couple of questions. The first is from Christina. Which is the firing frequency range of cholinergic fibers? Are levels of cholinergic release correlated with increasing firing frequency of the fibers?
That's a really good question. And I think that depends on the target region you're looking at. So we don't actually know within the basal forebrain. Definitely that's true. If you look at downstream brain regions like the visual cortex, we know a lot more about cholinergic projections to the visual cortex. But I think that's a really interesting question to try and like dissect out the cholinergic release properties in the like distal brain regions versus the ones locally. So we assume that to some extent they're from the same neurons, but it doesn't necessarily mean that like a firing frequency at 10 hertz or whatever results in the same release properties in the visual cortex or in the olfactory bulb or within the HDB. So that's one of the reasons that we think the sensor work is so interesting, because it lets us get at the actual like cholinergic release profile in different brain regions.
Great.
So yeah, the answer is we don't know.
So just one more question. It's from Alberto. He says, great talk. Is there an adaptation trial after trial in the timing or amplitude of the depression of ACH after the reward in HIT trials?
Yeah, so that is a really good question. And I didn't get to talk about any of the different sort of manipulations we did to the go-no-go task, but the answer is like with just sort of baseline learning of an odor pair, no, there doesn't seem to be much of a change in the slope or the suppression, either the amplitude or the timing of the suppression. But when you start to get like weird changes in the amplitude or the timing of things, or when you do things like pseudo learning, which is where you basically do the same task, but the availability of a reward is not linked to the odor presentation. So you actually see like pretty dramatic modulation of the post-reward suppression in that case, which I think is super weird because they still get the reward, but they respond to it totally differently if there's not the odor reward association. And also like reversal learning, you see like pretty interesting changes in the temporal profile, but hopefully we'll publish this in like the next month or so and that data will be in there.
Fantastic. Thank you, Elizabeth. So we'll thank Elizabeth once again, and thanks for the great questions. And we'll move on to the next speaker. So our next speaker today is Dr. Christoph Weidemann. Christoph is an associate professor at the Department of Psychology at Swansea University in Wales, and he currently does research at Columbia University's Department of Biomedical Engineering. And today, Christoph is going to be talking about neural measures of subsequent memory reflect endogenous variability in cognitive function.
Thanks for this, and thanks for the organizers. It's a great conference. I'll be presenting joint work with Mike Kahana, and I'll start off with a fairly obvious observation, although the theme is quite different from the previous talk. And just the observation that when we measure memory in the lab, there's usually a number of distinct phases in such an experiment. So we usually have a study phase where we ask participants to study a list of items. In the experiment that I'll be presenting today, this will be a list of words. And then there might be some period where they have to retain that information. And then later on, there will be some type of memory test where they have to retrieve the information and show that they encoded something earlier. And in the experiment that I'll present today, this will be a free recall task where they just simply, people just simply have to recall as many items as they can remember from the study phase in any order that they like. And performance in that memory task is a joint function of the processes that are involved at each of these stages. So when somebody successfully recalls an item, that requires that the item was successfully encoded and stored, and then later successfully retrieved. And obviously, a failure to recall an item could reflect a failure at each of these processes. So it's often quite hard to specifically attribute changes in performance in these memory tasks to individual components such as an encoding process. The way neuroscientists have tried to address this issue is by collecting ongoing brain activity during these tasks. So for example, we could measure brain activity during the encoding session and then analyze this activity as a function of subsequent performance in that later memory test. And so the idea being that any brain activity that distinguishes between later recall or failure to recall might be associated with encoding processes that then later allow this item to be retrieved. But there's obviously lots of different ways to measure this brain activity. And in this study, I'll be looking at EEG recordings, and this is just one example of what this setup might look like. And there's lots of ways to analyze this data. So from each electrode, we'll get a time series like this. And the way I'll be analyzing the data here is to decompose that signal into different frequencies. So this is a little cartoon of what this looks like. And we're looking at power at a range of different frequencies, from low frequency fluctuations to high frequency changes. And to just give you an overview of what the data might look like from such a encoding list, this is the beginning of a study list. We have just 3 items here. The words judge, star, and actor, and I've bolded these words, the word judge and actor here, to indicate that these words were later recalled, and star was studied but not later recalled. The associated brain activity is just plotted below. Time is on the x-axis, frequency is on the y-axis, and the colors indicate power at that time point, at that frequency. When you look at this, look at the neural activity this way, it's very hard to make sense of it and to see any consistent patterns. But by just aggregating across all items that were subsequently recalled, or all items that were not subsequently recalled, we can get a sense of to what extent brain activity distinguishes between subsequent recall. And I've illustrated this here so that you can see that if you aggregate all unrecalled items, that activity looks quite different from the activity for all recalled items. And you can also see that this change is quite sustained as a function of time. So the vertical line here is the onset of the item, and you can see this, these changes are quite long lasting. So from now on, when I'll be showing you these kind of data, I'm going to be aggregating across time and just show you power in different frequencies without resolving the time axis here. So it's very tempting to conclude by looking at this that, well, maybe this is a neural signature of encoding processes that determine whether an item successfully encoded so that it can be later retrieved. But an issue with that interpretation is that we don't have any direct control of whether or not an individual item is later recalled or not recalled. And indeed, there's lots of external factors that correlate with recall success. And so they present significant confounds in this analysis. And one of those variables is serial positions. This is just where in the study list an item is presented. I'm showing you this here. So what I'm showing you here is for individual items. We have 24 items in each study list. The probability that that item will be later recalled is a function of where in the list it was presented. And so you can see that items that were presented early in the list are much more likely to be later recalled than items that are presented in the middle of the list. And so when we calculate the contrast between recalled and unrecalled items, the recalled items disproportionately come from early list positions, and the unrecalled items are more likely to come from later list positions. And we can plot brain activity now instead of as a function of recall. We can plot brain activity as a function of serial position. And when we do this, we can also see marked changes in brain activity as a function of serial position. Just to remind you what the brain activity looked like as a function of recall, I've copied this here from the previous slide, and you can see that brain activity early on in the list looks very much like the brain activity for recalled items, and brain activity for later items looks very much like that for unrecalled items. And indeed, we can do the same kind of serial position analysis by just looking only at recalled items, and also by only looking at not recalled items. And you can see in each of those cases, serial position effects are evident in the brain activity. And so they might contribute to any subsequent memory contrasts that we're looking at. And of course, serial position is just one of many external factors that affect the likelihood of individual items to be recalled. So the goal here is to try to distinguish between the endogenous variability that is associated with encoding processes and effects of external factors such as serial position. So I'll start off with just showing you a multivariate way to calculate a subsequent memory effect. So what I'm doing here is I'm combining brain activity from all electrodes and across all frequencies to train a classifier, just a logistic regression model here, to predict the probability of recall for individual items. And then we can evaluate the performance of this classifier just by correlating the output of this model with the recall status of individual items. And so this is a bit of an unconventional way to evaluate the performance of a binary classifier. In fact, this is a point by serial correlation because recall status is a binary variable. But I'm doing this to be better able to compare this performance with the performance of the other models that I'll present shortly. And so across all the participants, we can then plot this correlation or the distribution of this correlation, and you can see that it's positive, and this is our subsequent, multivariate subsequent memory effect. And so now the next step is to try to calculate a corrected subsequent memory effect where we statistically remove the effects of external factors. And so here, instead of predicting the recall status of individual items based on brain activity, we now, as a first step, predict the recall status of individual items based on these external factors. And specifically, I'm considering recallability here of individual items, and I can talk to you later about how we estimate this. The serial position, list number and session number. So we have multiple sessions for each participant, and within each session there's multiple lists, and so there could be practice and interference effects, for example. So we throw all this into this model, and then we use brain activity to, rather than directly predict recall of individual items, we now predict the residual recall after statistically removing these external factors. And then we can proceed just as previously by correlating the output of this model with the residual, and then we get our corrected subsequent memory effect, which is slightly lower, but still quite substantial. And you can see that a large part of that subsequent memory effect remains even when we statistically remove the effects of these external factors. I'm going to skip over this part and move on to another way to look at this by now calculating list level subsequent memory effect. So instead of predicting the recall performance for individual items, we can also just look at recall performance for individual lists. That gets rid of some of the external factors just by aggregating across them. So every list has the same number of serial positions. So any changes in recall performance as a function of serial position will just get averaged out when we look at lists as a unit of analysis rather than items. And we can do the same thing now. We have a regression model here because we have the probability of recall across the entire list of items. And then we can correlate the output of that model with that probability of recall, and we get an uncorrected list level subsequent memory effect. And we can see that works quite well. We have a positive subsequent memory effect, but of course, there's still external factors that could contribute to that effect. And so I'm proceeding the same way that I did for the item level subsequent memory effect. We're accounting for remaining external factors by first predicting the list level recall performance on the basis of these factors. So looking at the average recallability for items, for all the items within the list and list number and session number. And then as a second step, predict that the residuals of that model based on the brain activity. And then again, we can correlate the output of that model with those residuals, and we get the corrected subsequent memory effect. And you can see again, just as for the item level subsequent memory effect, that that corrected subsequent memory effect is only slightly smaller and very robust. So just to wrap up, we can conclude that the substantial item and list level subsequent memory effect, even after accounting for external factors. We have relatively small effects of the external factors on the SME, even though we can see that these external factors can predict recall performance quite well. They don't seem to be a major confound in the contrast of neural activity as a function of subsequent memory. And therefore, we can conclude that the subsequent memory effect largely reflect endogenous variability that's associated with cognitive function rather than effects of these external variables. So the paper just very recently got accepted at the Journal of Experimental Psychology, Learning, Memory, and Cognition, and the preprint is available on my website at cogsci.info. Thanks very much.
Thank you, Christoph. That was a fantastic talk. I was wondering if you had like a kind of a prior manipulation or some words that were repeated more than others, would you expect the same result, or would you expect it to have an impact on the EEG?
Well, interesting. So, well, you could think of that as another external factor, right? If you repeat an item more than once, you essentially you could think of that as changing that item's recallability. So you're making some items more memorable in a way, and so you could model that the same way. I mean, it gets a bit tricky because you kind of it raises the question: are you only looking at how are you looking at the different the different encoding episodes, right? You could imagine that if you don't know that an item is going to be repeated, the brain activity at the initial exposure shouldn't distinguish between repeated and non-repeated items. But when an item is repeated, there's more going on than just the regular encoding process. You also recognize that the item was there previously. So I haven't done anything along those lines. I think you could probably model those effects in a similar fashion as to what I've presented here, but I think you'd have to think carefully about how to deal with those repetitions and the fact that that at the same time you're encoding that item, you're also retrieving the fact that you've seen it earlier in that same list.
Sorry, thank you.
No, thanks. Okay, well, thanks very much. I'm going to stop sharing. So,
okay, I guess we'll move on to our last speaker. Thanks again to Christoph. That was a really interesting talk. And our last speaker today is Dr. Mohammadi El-Gabi. Mohammadi is a postdoctoral neuroscientist at the Nuffield Department of Clinical Neuroscience at the University of Oxford. And he currently works in the lab of Tim Behrens, but will be presenting work from his previous postdoc with David Dupré. And today he's going to talk about a neural coactivity code for dynamic memory.
Thank you very much, and for this kind introduction. I'm going to be talking a little bit about the role of neural coactivity in encoding information and in supporting memory. And the sort of central question that we're interested in is what features of neural activity encode information. Now, consider this sort of toy example here, where you have a set of 6 neurons that fire differently to different stimuli, X and Y. So neurons 1, 2, and 3 need to fire more for stimulus Y, whereas 4, 5, and 6 fire more for stimulus X. So this is your standard sort of rate code that is sort of just throughout multiple species and systems. But then think of this second example here on the right, where now there's nothing about the single order or first order features of these neurons that encodes information about X and Y. However, if you look closely, you can see that neurons 1, 2, and 3 are highly coactive within a short time window in response to Y, whereas 4, 5, and 6 seem to be highly coactive or highly synchronous in response to stimulus X. So here the information about the stimuli is present as a sort of emergent feature or an emergent property of short time scale coactivity. So I'm going to be referring to this type of code as an emergent coactivity code. And this kind of this type of code has been sporadically reported in the literature in certain species and in certain systems, but the cognitive function that it might be playing has not been explored. And so our sort of sub-question that we're interested in is what are these emergent short timescale coactivity codes useful for? And we hypothesized that perhaps in situations where animals must form dynamic memories, that is, memories that are rapidly formed and flexibly retrieved, you might need to form or encode this information using short timescale coactivity, given the speed of both stabilizing these coactivity codes and also retrieving them. And sort of to further clarify what I mean by dynamic memories, let's just jump straight into the task that we're using. I designed a task for mice in which mice have to learn two new behavioral contingencies every day. And the contingencies look something like this. So when a tone comes on in the presence of one set of LED displays, the animal needs to go to one of two dispensers, in this case dispenser 1, in order to get sucrose, and needs to avoid an identical looking dispenser in a different location, which delivers quinine, which is quite a bitter substance. And then in the presence of the same tone, but when a different set of LED displays is illuminated, now the contingency is reversed. So dispenser 2 is now the good one, the one that delivers sucrose, and dispenser 1 delivers quinine. So the animal has to do to approach a different dispenser. And the animal learns a new example of this every day, and we can show that the animal starts off naive. On a given day, the structure is as follows. First of all, animals will explore the learning enclosure in the absence of any tones or sucrose or quinine, but in the presence of the LED displays, and also explore a control enclosure. And then it receives 2 learning sessions in each contingency. And then finally, after the last learning session, an hour later, the animal is tested in a memory probe test in extinction. So the tone is delivered, but no sucrose or quinine are given. And crucially, in that probe session, while the animal is in the same enclosure, we pseudo-randomly switch between the 2 LED displays. So the animals have to quickly update their behavior to be in line with the contingency associated with the LED displays. And so this satisfies both of the criteria that we've set for a dynamic memory task. Animals have to learn it rapidly every day within a couple of hours, a few hours, but also they have to retrieve it dynamically because we shift between these LED displays. And what we see is the animals indeed do remember or retain a memory of this contingency discrimination into the probe test. And this is sort of a pictorial representation of roughly what they're learning in this task. But this is nice because it gives us a dynamic memory paradigm in which we can now test hypotheses about neural mechanisms. And for various reasons, we thought that dorsal hippocampus might be important. So we recorded from dorsal CA1 as the mice performed this task. And we could record from multiple individual neurons. Here I'm going to talk about data from principal or putative principal neurons. And crucially, in order to get at coactivity, we can detect patterns of coactivity using a method that relies on independent component analysis to isolate these coactivity patterns displayed here in these stem plots, in which we use these sort of weight vectors to display the weight of each neuron that's been recorded in the population to a given coactivity pattern. So this is one coactivity pattern, this is another, and so on. And this allows us to do multiple things. It allows us to track the strength of these coactivity patterns across time with a high precision, as you can see in this blue plot here. It allows us to also see which patterns are detected in which sessions, but also we can isolate the highly contributing neurons, so the neurons highlighted in color here in blue or here in red, and see and look at the properties of these neurons. So one of the first things that we did was to see if there are patterns that are present in one contingency and not the other. And so if you look at this similarity matrix here, you can see that highlighted in blue is a pattern that is present in both contingencies, which we're going to refer to as contingency invariant, whereas highlighted in orange is a pattern that is only present in one of the 2 contingencies. And so we can isolate these 2 types of patterns, and sort of their similarity profiles are shown here on the right. So we do see some short time scale coactivity patterns. Crucially, all of these coactivity patterns that I've talked about so far have been detected in a short time window, in a 25 millisecond time window, which roughly corresponds to the membrane time constant of these principal pyramidal neurons. And so, so we can see some contingency discrimination in these connectivity patterns in CA1. Now, the next question that we asked was, is whether any of these patterns, or whether these contingency discriminating patterns are indeed showing an emergent coding that is a function of their connectivity. In other words, are the properties of the individual neurons somehow also correlated with contingency? And so, as a sanity check, we first of all just check that the sort of Pearson correlations between neurons within a given connectivity pattern are higher in the preferred contingency, and that is indeed the case. But now we were interested in this first order properties. So, are these neurons also disambiguating contingency by their rate or other first order property? And first of all, we looked at their firing rate across contingencies, and that's not different. We also looked at their firing rate at the correct and incorrect location, so the sucrose versus quinine, and that's not different either. There's no relationship between their firing rate and distance from the goal. But also, even when we look at the single neuron temporal properties, so for example, when we look at the interspike intervals distribution across contingencies, that's not different either. And the temporal tuning to the actual trial stages themselves, so the onset of the tone, the onset of drop delivery is not different across contingencies. And even when we look at their tuning to theta cycles, so the theta phase coherence of these member neurons is not different across contingencies. So basically, every single neuron measure that we've looked at so far shows that there is no coding of contingency at a single neuron level. It seems like this is an emergent coactivity code for behavioral contingencies. And so this looks something like the schematic that I showed at the beginning, but now instead of sort of raw stimuli, we're looking at something a bit more abstract, sort of these contingencies. So we then asked whether this contingency discrimination somehow helps the animal perform in a task or has any relationship with task performance. And to do this, we simply track the expression strength of these connectivity patterns during exploration and learning. And what we see is that contingency invariant patterns are already increasing in strength during exploration, whereas contingency discriminating patterns seem to only robustly increase during learning. And a similar pattern is seen if you look at the reactivation of these patterns in sharp-wave ripples during sleep. So the sleep sessions either side of exploration, you see a reactivation of contingency invariant patterns, but not contingency discriminating ones. But the contingency discriminating patterns seem to be reactivated selectively in the sleep sessions after learning. So together, this data from both the online behavior and the offline reactivation suggests that contingency discriminating coactivity develops with learning. We can also relate it more directly to performance in the probe, and we see that contingency discriminating coactivity is stronger before correct compared to incorrect trials. The same is not true for contingency invariant patterns. And interestingly, if we look at longer timescale coactivity patterns that are contingency discriminating, while we can see contingency discrimination at these longer timescales, these patterns are not reinstated in a probe in a manner that predicts performance, suggesting that this is a particular feature of these short timescale coactivity patterns. And then finally, what we were interested in is to see whether these contingency discriminating coactivity patterns have any causal relationship with the actual behavioral performance of the animals. And so in order to do this, we silenced inputs to the CA1 coming from the left, CA3, which previous work has shown that these inputs are highly plastic. And we reasoned that if we silence them during the learning phase, we might disrupt contingency discriminating coactivity patterns from developing. And that is, sort of long story short, that is exactly what we see, despite so basic features of coactivity not being affected. And so we see this by looking at this using this ICA method, but we see this also using a different method that relies on pairwise correlations and Bayesian classification. And so we can get a mechanistic handle at these contingency discriminating coactivity patterns and see whether behavior is also impaired. And bearing in mind that we're silencing only during learning, but what we see is that during memory performance in the probe, the animals basically go to chance when you get rid of these patterns, suggesting that they may be necessary for this memory performance. If you silence the control pathway coming from the right, you see that you don't impair performance. And then finally, if you silence the same pathway, but in a one contingency learning task, you don't impair performance. So all of this together, I realize the time is up, but all of this together, I'll come back to this summary slide, but all of this together suggests that these contingency discriminating coactivity patterns are necessary for dynamic memory performance. And in the end, I would like to sort of thank all of the Gray Lab, and especially people who are involved in the study, including David, Haley, and Vito. Thank you all for listening.
Thank you so much. That's really impressive work. I hope I'm unfortunately a bit out of my depth, so I hope there'll be some questions from the audience. But maybe could you talk a bit more about the mechanism of that blocks the coactivity patterns?
Sure. So the silencing of left CA3 inputs. So we previously, both myself and others in the Paulson lab showed that these inputs coming from the left CA3 show robust long-term potentiation, long-term depression. And so we reasoned, because these patterns seem to develop during learning, we reasoned that if we interfere with inputs that seem to be plastic, and also inputs that seem to be important for certain types of long-term memory, we might selectively interfere with them, and that's exactly what we find. These patterns are so this is this. This is this slide here, where we show that the distribution between contingency similarity of the independent components, or the coactivity patterns, is shifted in the light-on condition, shifted towards contingency invariance. So suggesting that we have indeed disrupted these contingency discriminating patterns.
Fantastic, thank you. So we have a question here from Daniel. It says, coactivity coding sounds super interesting. Could you briefly explain the difference to phase coding?
Right, sure. So phase coding would suggest that there's something about the single neuron properties. So something about the locking of the spikes of a single neuron to a phase of some oscillation that tells you what stimulus is on. So let's say you've got an oscillation at the peak. If you fire at the peak, then stimulus X is on. If you fire at the trough, then stimulus Y is on. So that would be phase coding, and it's a single neuron code. Although you can imagine that it could be a coactivity code too. But the key difference is that a phase code doesn't have to be an emergent code. It doesn't have to be a property of multiple neurons. It can be a property of just one neuron. Whereas coactivity code is, the information is only present if you look at the combined activity of multiple neurons.
We might try and squeeze in one more question from Nick Robinson. What happens if you repeat the analysis with 120 millisecond time bin? Do you see this occurrence of these discriminating events, particularly at a certain theta phase?
Right. So I think there's two questions there. So we do 120 millisecond. If we do longer bins, you can still detect contingency discriminating patterns. Although, so when I try 1 second, these patterns are not reinstated in a way that predicts performance. I haven't looked at sort of everything in between 25 and 1,000. The other question was about, sorry, was it about the phase of?
Yeah, we may not have time to answer it, but do you see the occurrence of these events at a certain theta phase?
Right. So there seems to be a slight bias, but not a statistically significant one, towards the members of these contingently discriminating patterns happening slightly earlier in the theta cycle, or firing slightly earlier in the theta cycle than the invariant ones. But it's a very small bias.
Well, thank you so much. That was a really interesting talk, and thank you to all of our panelists. I think we had a really good session.
Thanks, Elaine, for emceeing.
Thank you.
די, אל תשלחו לי עכשיו. תודה. תכף אני אתקשר לכם, אני אוהבת אתכם.
One more recommendation is to mute yourself when the other person is talking. This generally, like it seems in YouTube, people see the person who talks. So even if you have some kind of sound, your face pops up suddenly. So we're just trying to avoid that. Okay, I think we can get started. So welcome everyone. My name is Shankar Alagappan, and along with me I have Dan Wessling running the session. We are excited to hear from three speakers. They'll be talking on the topic of how structure of tasks is processed in the brain. The format of the current session is a traditional talk, which means the speakers have over 12 minutes to present their work, and followed by about 3 minutes for question and answers. I highly recommend using the Q&A panel available in Zoom, and also to vote for your favorite questions. And please feel free to use the chat to interact with the panelists and other attendees. I hope you have been having a good time at Neuromatch, and do let everyone know about the sessions using Twitter or any other social media that you'll be using. So without further ado, we'll be bringing up our first speaker, Maria Eckstein from University of California, Berkeley, who will be talking about hierarchical learning in complex action sequences. The floor is yours, Maria.
Hello, thank you so much. Like you just said, I'm Maria Eckstein, and thank you so much for having me. I'm doing my PhD with Ann Collins at UC Berkeley, and I'll tell you about a project that I'm currently working on with the title, How the Mind Creates Structure, Hierarchical Learning of Complex Action Sequences. I want to start by saying everyone agrees that we use hierarchy. For example, in the field of machine learning, there's a famous example of the 4 rooms domain. Basically, you have an artificial agent that is dropped at a random location in this blue playing field, say the green circle here, and it needs to find the red cross up there by trial and error. Trial and error means that the agent can pick one action, up, down, left, or right, on each trial, and then observe what happens. It can't look ahead. All the dark blue tiles are walls that the agent cannot cross. What happens in this case is that a standard non-hierarchical reinforcement learning agent will often just get stuck in the room in which it spawned and not find the red cross before the time runs out. Now, an easy fix for this problem is to add hierarchy. When we add the action, go to the door, to the agent's repertoire, all of a sudden, it will be able to explore all 4 rooms more evenly and find the red cross. This simple example is supposed to highlight how seemingly complex problems can all of a sudden become very easy once we equip an agent with hierarchy. In this case, we use temporal abstraction, which is the ability to execute multi-step actions. Lots of research in machine learning has used hierarchy to solve complex problems, ranging from the options framework to MaxQ, pseudo reinforcement learning, MAML, R-squared, option critique, to name just a few. In psychology, research has been interested in hierarchy for several decades now. One famous example is the case of expertise. For example, in chess masters are able to remember the positions of dozens of pieces on a board, and it turns out their trick is state abstraction, another form of hierarchy. Basically, chess masters chunk constellations of several pieces into a single mental representation to save memory space and be able to remember and plan more efficiently. recent research has put increasingly more focus on formalizing hierarchical cognition. And many researchers have proposed hierarchical Bayesian models like Josh Tenenbaum, Tom Griffiths, and more recently, many hierarchical reinforcement learning models have come up. For example, by Matt Botvinnik, Michael Frank, David Bader, and we also have a new paper that's coming out in PNAS this year. In neuroscience, there's strong evidence that the brain is organized hierarchically as well. The prefrontal cortex exerts control over subcortical regions, and in addition, the control loops between prefrontal cortex and subcortical regions themselves are organized hierarchically, such as more anterior loops operate at more abstract levels than the posterior loops. So it's not that everybody agrees that we use hierarchy, but how do we create hierarchical representations? Like in the 4 rooms domains, once we have the right hierarchical representation, problems are often easy to solve. But where does this representation come from? Some computational studies show that humans use a Bayes optimal task decomposition, but finding this decomposition usually requires full knowledge of the state space and lots of processing power and time, and humans usually don't have access to either. What we propose here is that humans create hierarchical representations piece by piece. We continuously learn new, ever more complex actions that we then add to our action repertoire. Say we are born with a very basic set of actions, like stretch your arm, move your fingers, wave your hands. This baby here might explore the world around him by randomly executing one of these actions at a time. At some point, he accidentally hits the rattle and it makes a sound. This new sound triggers the baby's curiosity. What happens next is that the baby wants to produce that sound again. He sets a goal for himself and is intrinsically motivated to reach it. Over time, he will learn how to grasp the rattle and make that sound, and he'll add this new skill to his action repertoire and use it for future exploration. In this way, we don't require a reward at every time step in order to learn something useful. We can learn continuously just by following our intrinsic motivation, which is shaped to our environment. Over time, our action repertoire expands and gets more complex, and for this violinist here, Andrew Bird, playing an entire song might just be one action in his repertoire. To test this, we came up with the following task. Participants see this machine and are told that their goal is to make stars. The machine makes a star when they type in the correct sequence of 4 keys. Say we press the orange key first, nothing happens yet. Then we press the blue key, and a gear appears in the machine. This is exciting. It's not the star we were hoping for, but at least something happened. The gear is the rattle sound for the baby. It motivates us to learn the subsequence and use it for further exploration. So this is what we do next. We press yellow again and blue again, and indeed we can get the gear back. Now let's try something different. This time we choose the purple key and the red key, and we get a red light. Same story, we want to make it work again, and it does. We now have added 2 new actions to our repertoire, the gear and the light. Now let's see what happens when we do both, the gear and then the light. Orange, blue, gear, purple, red, light. And the combination of the gear and the light produces a star. In the task, participants had to learn how to make 4 different stars, each with a different 4-key sequence. Crucially, each 4-key sequence was composed of the same 4 2-key sequences, including the gear and the red light. Participants first had a learning phase that looked basically like what I just showed you. Participants are told to make a specific star and get points whenever they got this star. After 400 trials of this, they do a transfer phase. What happens here is that all of a sudden, the learned rules stop working. We have 2 different conditions for the transfer phase, low and high. In the low transfer phase, we change the low rules, i.e. which keys make a 2-key sequence. In the high transfer phase, we change the high rules, i.e. which combination of 2-key sequences makes a star. If participants learn the game in a hierarchical way, low and high transfer should affect performance differently. Of course, we counterbalanced everything, for example, the assignment of keys to colors, key sequences to items, order of low and high transfer blocks, etc. We've collected 60 participants so far. So let me show you how participants did this task. I'm plotting the trial on the x-axis and accuracy, which is whether they managed to get the gold star, on the y-axis. In the beginning, participants don't get the right star, but they learn pretty fast and get up to 20% at the end of the first block. Then the gold star changes, and they start around chance again. At some point, stars repeat and participants get better and better over time. Then the transfer phase starts, and we switch out the rules, and participants are back at 0. But they recover fast and relearn the new rules pretty well. What we did next was to go through our hypotheses one by one and check each. First, we wanted to know whether participants would set sub-goals and be intrinsically motivated to execute the 2-key sequences, even though they didn't get a proper reward for them. What I'm showing here is trials on the x-axis, and I aligned them to the first time a participant executed a particular 2-key sequence. On the y-axis, I'm showing how often participants use this same sequence again. The blue dots are valid sequences, the ones that lead to a gear or light, and And the red dots are invalid sequences, which do not lead to anything, but have the same structure other ways. What we see is that participants were more likely to repeat valid sequences than invalid ones, as if they were motivated by the signal to repeat and practice these sequences. In the plot below, I'm showing the 25 trials of just the first block on the x-axis and the frequency of valid and invalid sequences on the y-axis. We see that in the beginning, valid and invalid sequences are equally likely. But over time, the valid sequences become more and more likely, as if participants make them their basis for exploration once they discover them. Importantly, these analyses would be totally confounded by rewards if I left the trials in there in which participants actually got a star. So I removed all rewarded trials here, and the patterns still come out. Next, I wanted to know if participants really created new actions in this game, if they would start treating valid 2 key sequences as distinct actions. If that is true, participants would show a specific pattern of response times. At the beginning of a trial, they would think about which sequence to use, then execute both keys of that sequence in one swoosh, then think again, and maybe execute another sequence. This is exactly the pattern that we found. I'm plotting the 4 keys of a trial on the x-axis here, and the response times for each key on the y-axis. Each color is 1 participant. Participants took their time to press the first key in a sequence. The second key came really fast after that. Then they think again at the beginning of the second sequence, and the last key comes very fast. This looks like participants select 2 unique key sequences and not 4 unique keys. Lastly, I wanted to look at the transfer phase and see if you have differences between the high and low condition. As a reminder, in the low condition, we changed individual keys in the 2 key sequence. In the high condition, we changed a combination of 2 key sequences that make a star. What I was interested in was whether people relearned the low-level 2 key sequences as easily as the high-level sequences of sequences. Our hypothesis was that the lower level would be more consolidated and less accessible. Basically, once a key sequence becomes an action, it would be hard to change it. We thought that the high level would be much more flexible and malleable because it had not yet been consolidated. And it looks like this is exactly what happened. I'm plotting only the transfer phase here, trials on the x-axis and accuracy on the y-axis. High transfers in red and low transfer in blue. And we see that participants relearn much faster in the high transfer phase than in the low transfer phase, and also show faster response times here on the right. This suggests that participants chunk 2 key sequences into actions, and that these consolidated actions are harder to unlearn. So in conclusion, there has been a tremendous amount of research on hierarchy over the past decades, and everyone agrees that it is beneficial and can, theoretically, solve some of our hardest problems. Nevertheless, we don't fully understand yet how humans create hierarchy. We propose that what we do is to expand our action repertoire by adding more and more complex actions, and that we use non-rewarding signals from our environment to achieve this. We have behavioral evidence that humans indeed chunk action sequences into distinct new actions and use them as a basis for further exploration. What I want to do next is to create a hierarchical reinforcement learning model to formalize this idea and explore our dataset in more detail. And with this, I would like to thank all of my collaborators on this project. First of all, my advisor, Ann Collins, and all members of the CCN Lab at Berkeley. Please get in touch if you have any questions or comments. My email is maria.eckstein@berkeley.edu. Thank you so much.
Thank you, Maria. This is the applause that you probably heard from attendees if you were in a live presentation. But we do have some questions now. So the first one is from Eleanor. This might be a very general question, but do you have any predictions regarding the neural basis of hierarchical coding in the brain? Where and how would it be encoded?
Yeah, that's a fantastic question. And I'm just starting to scratch the surface of this. So there's a lot of research, both in humans and in rodents and in non-human primates, about the neural representation of action sequences and of hierarchical action sequences. And from what I've got so far, it seems that the basal ganglia play a really important role there for both learning and execution of those hierarchical sequences. And there's this study by Geddes et al., I think in 2018 maybe, where they do some optogenetic manipulation and show, they really go in depth here of showing how those action sequences are represented. I can maybe put that paper in the chat if you're interested. But yeah, there's a lot of research there. So it's like mostly basal ganglia, and then I think some interactions with the prefrontal cortex in the corticobasal ganglia loops.
Great. So we have another question. How do you know that people weren't learning hierarchically simply because your task encourages it?
Yeah, that's a very good question. I think that's always the debate. How much do you put into your task and how much comes out? So I think I definitely designed the task so that people would be motivated to learn hierarchically. And then I'm interested in how they create this hierarchy over time. So the idea is that we don't put rewards, we put some kind of information signal at the end of those two key sequences to see if that helps people learn them. A control that I've been wanting to run is basically to take away those signals, to take away the red light and the gear, and to see what happens then. Are people still able to infer the hierarchy, even if you don't give them this cue along the way? And my hypothesis is that people would still be able to learn it, but it would take them much longer. So the idea here is that we can kind of bootstrap our hierarchical learning if you pay attention to what signals the environment is giving us, and if we're intrinsically motivated to kind of learn some control over the environment as we go.
That's a wonderful answer. I think it kind of answers the next question, which is, do you think the formation and relearning of two key sequences is contingent on the feedback signal?
So is that in the transfer phase? I'm assuming the relearning of the two key sequences in the transfer phase? Is it
transfer phase, yeah.
Okay.
Or maybe even, yeah, I don't know if it is between these switches or between the transfer phase. Yeah. So each time you see there is an increase in performance and there is a drop because there is a change in the contingency.
Yeah. So when in the learning curves that I showed in the beginning, that keep going up and down, that is basically because participants are told in the beginning of each block which star they are supposed to reach in this block. So say the orange star, and then there's a green star, there's a blue star. So the reason it drops is that people might still transfer over all the two key sequences that I have previously learned, but they still need to remember or relearn how to combine those together.
Okay, and is it valid for the transfer phase also?
Yeah, in the transfer phase, I think it's really important that people get this feedback. In the low transfer phase, when we change the two key sequences, I think it is important for people to get that feedback. And all of a sudden, when they press the two keys, the gear does not show up anymore. So this is an important signal for people to relearn. But I haven't looked into that into too much detail, so that's going to go into my to-do list.
Yeah, so I think we are at the end of our time. So there is one more question, but can you quickly check and answer it in the chat?
Yeah, I'll do that.
So we'll be moving on to our next speaker. We have Leonie Glitz from University of Oxford, who will be talking about how representations adapt during probabilistic learning. Leonie, the floor is yours.
Thank you. 1 second. Okay, cool. Hi, my name is Leonie. I'm a second-year PhD student at the University of Oxford, supervised by Helen Barron and Chris Summerfield. And before I get into today's talk, I just want to make it really clear that this is very much a shared first author work with the fantastic Neil Garrett, as well as a collaboration with Keno Yuchums and my supervisor, Chris. Okay, so to start off, I want you to imagine that you're a bank robber, and what you do on a daily is rob a bank. Right, but in your town, banks work in a specific way, whereas once you enter their lobby, they have a security system where you always see two doors, a light door and a dark door. Now, each of those two doors has a certain probability of either leading you back to the exit, so you leave the bank, or leading you to the vault where the money is kept. And one door will always be more likely to lead you to one outcome state and the other one to the other. You have to learn these transition probabilities by trial and error. And importantly, in this task, you shouldn't learn the value function, but you should actually learn the transition function. Because on half of the trials, or I guess half of your days that you spend robbing banks, the police has previously been informed that you're going to rob this particular bank. So you should really try your very best when the police knows that you're coming to choose the door that is going to lead you back to the exit to avoid losing money and being punished. Now, let's take a step back based on what I've told you so far and think about what sort of neural structures could be involved in the coding of this task. I think probably for a lot of you, a lot of structures will come to mind, but one that I want to highlight in particular here is the hippocampal formation. So we know a lot about the hippocampal formation generally, and one of the things we know is that the hippocampal formation and its surrounding structures really seems to care about the distance between states, for example, on an implicit map, as beautifully shown in a paper led by Mona Garvard in 2017. And also that this region really seems to care about tasks that are more similar to what I've just shown you. And this is a paper by Ari Borman from 2016, where he showed that the hippocampal formation encodes the surprise that you have at reaching a certain outcome state. So it seems to encode transition probabilities from states to outcomes. Now, this is great, but actually in the real world, we very often don't just have one task that we want to solve, but we have a lot of different tasks that might have different sort of transition contingencies. So a question that is still largely unanswered is whether the hippocampal formation is also the part of the brain that allows you to generalize across contexts when you can do so, and if it is, how this is implemented. So to illustrate what I mean, now imagine that actually in your town, rather than just having one bank, you have 4 different banks. To help you, you have some information about those banks, whereby you know that, for example, here, the blue and the green bank or context always have identical transition probabilities. Now we refer to these as dependent blocks. And in these dependent blocks, because you know that the transition probabilities are identical, you should really share information or generalize information across the 2 contexts that you encounter in these blocks. In contrast to that, in what we refer to as independent blocks, the 2 contexts that you encounter are not correlated with each other. So they're neither correlated nor anti-correlated. So in these blocks, you should really do your very best to keep the information you gather regarding the transition structure separate between the 2 contexts. So this is then how our task allows us to address the question I just phrased, which is whether the hippocampal formation allows us to generalize across contexts when it's appropriate to do so. So here in our dependent blocks, but also allows us to keep representation separate when it's inappropriate, like in our independent blocks, and also how this is done. Just a quick sanity check before we go into the neuroimaging analysis, which is obviously needed to address this question. Before we do this, we have to check that our participants have understood the task. So what we did is we told them basically exactly what I just told you. And we then can check whether they've integrated information behaviorally as is appropriate. So what you can see here is that participants used information given on the previous 5 trials from both the currently visible context, shown in blue, and the other context pretty much equally to inform their choices. And in contrast to this, in the independent blocks, where the underlying transition probabilities of the 2 contexts were not related, they only used information from the context they were currently seeing that they'd gathered on the previous 5 trials to predict choice. This is great. This is what we wanted. So to then give ourselves a more specific estimate of what was going on, we then fit a computational model to the data. Well, we fit a lot, but we decided on one. Really briefly, our final model was, as appropriate for the design, a mixture model where we had a free parameter that allowed us to adjust the amount of integration between the information that you get from the current context and the not currently visible context. And that amount of integration was allowed to differ between the 2 conditions. And just really quickly, if you use that model to then simulate participants, this also provides quite a good qualitative fit to our data, here shown in black. Cool. So now that we know this, we can get back to our actual sort of neuroimaging question, which is whether the hippocampal formation allows you to generalize across contexts when this is appropriate and how this is done. The first thing we then need to ask is whether in our task, like in previous tasks, we actually observe any sort of encoding of transition probabilities in the hippocampal formation. To do this, we ran a univariate analysis where we took the unsigned state prediction error from our model. And the state prediction error really briefly is just the difference between a transition you expected and a transition that you actually experienced. So a surprise signal, if you will. We took that unsigned state prediction error and we looked at where in the brain the bold signal, this was done in fMRI, the bold signal most strongly covaried with this state prediction error. And what we found was a bilateral medial temporal lobe cluster, which included parts of the hippocampus, entorhinal cortex, and amygdala, which is great, right? So this is what we'd sort of expected based on previous research. And we find this. Fantastic. This means that we can now check whether this encoding of transition probabilities in the medial temporal lobe varies depending on whether you want to generalize across contexts or whether you do not want to do this. To address this question, we used predominantly one method, which is a multivariate analysis method called representational similarity analysis, where you're essentially just looking at how different the pattern of voxel activity in your region of interest is between two different contexts. And just as a disclaimer, what I'm showing you today is predominantly within our state prediction error mask, so a medial temporal lobe mask. We looked at this at two different time points, the first one being here. So basically what happened on each trial is that participants would first see one of the two doors, but not be allowed to indicate their choice. And we basically looked at this door onset period to see if we could find any sort of predictive transition signals and whether those were represented differently between the contexts in the two conditions. Before I get into that, just a reminder as to why we've done what we've done. So the transition probabilities varied throughout the experiment. And to be able to compare identical transition probability bins from this door that was first presented to the risky state, we had to bin trials into transition probabilities because especially in the independent blocks, these would not be occurring at the same time. So we binned our trials into 4 sets of transition probabilities. And to assess whether the similarity of these predictive transition representations differed between conditions, we compared the representation of identical probability bins, here the diagonal, identical probability bins, between the two contexts that you saw in each of the conditions, compared to how similar the representation of non-identical probability bins was between the two contexts that you encountered in each condition. So here our hypothesis was that this difference between identical or between the similarity of identical probability bins between the two contexts and non-identical probability bins between the two contexts, that this difference would be greater in the dependent condition than in the independent condition. Because in the independent condition, you don't want to encode probabilities in a similar way between the two contexts. And this is what we find. So in the dependent condition, we find that identical probability bins are represented significantly more similarly between the two contexts that you experience in those blocks than non-identical transition probability bins. We also find that this is not the case in the independent condition and that there is a significant difference between the two. This is great. So this indicates that yes, it seems like the medial temporal lobe encoding of our state transition structure varies depending on whether you want to generalize across contexts, like in the dependent condition or not. And specifically, this seems to vary in terms of varying the similarity of the prospective representations that you need at the time of planning. Now you might say, well, maybe this is not a property of hippocampal encoding, but maybe this is some sort of similarity in representations that arises as a function of planning at the time of planning. To check this, we also looked at a second time point, so a time point when participants are given feedback regarding which outcome state they'd gotten to. And here we used a very similar sort of setup to ask whether the similarity of action outcome state associations in our medial temporal lobe cluster differs between conditions. So what we looked at here more specifically is whether if you in context 1 choose a specific door, for example, the light door, and get to a specific outcome state, for example, the risky state, whether how similarly that is encoded to when you get the same sort of action outcome state feedback in the other context in each condition, which is again the diagonal, and then we compared that to the similarity in encoding of non-identical pairings of action outcome state associations. Again, we had the same hypothesis where the diagonal off-diagonal difference would be greater in the dependent than the independent condition. And this is what we see. Again, with the dependent condition, similarity on the diagonal being significantly greater than the off diagonal, and there being no difference in the independent condition. Because this effect looks a little bit weaker, and to convince ourselves and you, we also ran a whole brain searchlight analysis here, where we looked at where in the brain the difference between the diagonal and the off diagonal is greater for the dependent and the independent block. And what we found here, sorry, I'm almost done. What we found here is a bilateral hippocampus cluster that extended down into the pons. This is great. It means that this encoding also varies depending on whether you want to generalize or not at the time of feedback in the medial temporal lobe. So then to wrap up, what is the take home? We were asking whether the hippocampal formation allows you to generalize across context when this is appropriate and how this is done. And what we found is data that suggests that the hippocampal formation is what allows for flexible levels of generalization by adjusting the pattern similarity with which transition models are encoded or updated at the time of feedback and retrieved to aid planning. And I'm happy to discuss how this links into previous research during the question time, because I'm aware I'm over time. Thank you.
Thanks for the wonderful talk. As we are waiting for questions, I'm not very familiar with this type of research, but I'm curious, so there are people with epilepsy who often have lesions, or they have some kind of issues with the hippocampal regions, right? So in those patients, do you think there would be a deficit in this kind of learning? Has there been any evidence for that?
So I know that patients with hippocampal, I think dorsal hippocampus lesions have been shown to be impaired in model-based planning. So there's a paper that's out this year by someone whose last name is Weekblad, I think. So they show impairments in model-based planning, but I'm not aware of any studies looking at sort of generalization across different task models in these patients, but they exist. So that would definitely be something that would be interesting to do.
Got it. So yeah, I mean, just to remind the attendees, you can ask your questions in the Q&A box. In the meantime, so where do you think you're going to take this research next?
I think for now we're aiming to get a preprint out.
Okay.
But
fair enough.
I think in the future it'd be really interesting to see. So I don't know if this was clear, but for example, it would be really interesting to vary the contexts, or to change over the contexts that are assigned to what are dependent blocks and independent blocks. So to basically have established this block structure and then mix them up and see if even across different blocks in terms of temporal order, there would still be this correlation structure in terms of encoding, or whether it's some function maybe of being in this fixed structure. But that's just one idea.
Yeah, very interesting. So if there are...
If you have time.
Yeah, go ahead.
Because I don't seem to have access to the question and answer field. So you talked about some computational models that you fitted to that, and I was interested in what these look like.
Yeah, great question. Sorry, I glossed over that for time, but yeah. So basically what we did is we have two sets of transition probability estimates. One set where you have 4 separate estimates for each of the contexts, so one for each of the contexts. And then we have a combined transition probability estimate that gets updated on every trial, irrespective of which context you see. And then we have a free parameter that we call W that integrates between those two sets of transition probabilities. And we allow that W parameter to basically flip between block types. So if in both conditions there was an equal amount of integration of information, it would be 0.5 in both, so it wouldn't matter. But what we find is actually that participants do integrate or do use the combined estimate much more in dependent blocks than in independent blocks. Sorry, I hope that helps. That's a bit rambly.
All right, thank you again. Next, we have our final speaker for the session, Dr. Shirley Mark, who will be talking about how structural knowledge is transferred across different cognitive maps. Take it away, Shirley.
Okay, hi. So I'm Shirley. I'm working with Tim Burns, and I'm going to talk with you today about transferring structural knowledge across cognitive maps in humans and models. So what do I mean by structural models? So imagine that you're looking at the shape of the moon today. You can immediately infer what is going to be the shape of the moon tomorrow. And you can do that because you know that the shape of the moon follows a periodic cycle. And you can characterize this by using graph in which each node on the graph is the shape of the moon, and two shapes can appear one after the other if they are connected by a link, by an edge. But the shape of the moon is not the only thing that follows a periodic pattern in nature. This is quite a prevalent pattern. And we know that there is also the cycle, that also the season cycle follow this periodic cycle, periodicity. But these two circumstances are really different from one another. They're different in the stimuli, but not only that, they're different in the size of the graph. But nevertheless, all of us as humans can say what is the commonality, that these two circumstances or phenomena follow this periodic pattern. Okay, so if relations between elements follow a common prevalent pattern, how can we generalize and exploit this knowledge? So there are other types of regularities that appear in nature. For example, we have the community structure, in which a node within a community tend to be more connected to another community, another node within that community compared to a node with a different community. And this regularity can appear in different graphs that have different shapes and a number of nodes, different particularities. And we can call this family of graphs that has the same type of regularity as a structural form. And then each structural form is a family of graphs that can have different number of nodes. And there are more types of structural forms, such as grid and trees. But in this work, I will focus in two types of structural form, the community structure and the grid, and I'll try to answer how can the knowledge of structural form be efficiently represented to a large generalization, and how can we benefit from abstract structural knowledge, and I'll use the HMM, the Intel Markov model framework for that. And then I will show two behavioral experiments that show that people can represent and transfer such abstract structural knowledge. And then I will show you a preliminary fMRI result that shows that this abstract and flexible structural knowledge is encoded in the MTL. Okay, so our approach for answering this question is by a graph learning task in which two stimuli, in this case two pictures, can appear next to each other if they are connected on this graph. But of course not the agents and participants can see the bird view of these graphs, and they learn the graphs using associations. And for generalization, the important thing, the first thing that we need is that we need a separate representation for the structure of the task and the assignment of the stimuli. And for that, the HMM framework is really good because it has two different representations for the structure and the stimuli. We have an abstract transition matrix that tells us which state can follow one another, and we have an emission matrix that tells us which stimuli belong to which state. So presumably we can store abstract transition matrix and then infer which matrix we need to use when we encounter a new task rather than learning it. But this is not really good enough because I told you before that within a structural form, the size of the graph, the number of nodes can be different. So this entanglement, this separation of sensory stimuli and structure is not enough. So to solve this problem, we approximated the transition matrix using a basis set, which is a set of vectors which can be used to compress and flexibly construct the transition structure. And importantly, they can be stretched and compressed, so we need just one basis set for each structural form. So what we do is we hold these basis vectors for arbitrary large enough graph. So in this example, we have the hexagonal grid structure, and this is examples of these two vectors. And when we encounter a new task, we need to interpolate each vector according to the currently estimated graph size according to some prior. And then we can estimate the transition metrics. And we can do similar thing also with the community structure. So now we can think about it as a kind of a generative model of graphs, in which graphs are generated from these basis sets. And every time we encounter a new task, we need to infer what is the correct structural form, the size of the graph, and learn the emission metrics. So using such agent, we can infer correctly the structural form and also the dimension of the graph. But what can we do? Why is it good to transfer the transition structure rather than learning it? So for example, if we do have this transfer, we can infer unobserved relations. So if we give the agent to learn this hexagonal graph, but in this case, the agent doesn't see all the red edges that you see over here. So if we learn, and now we are going to give him the following task. We are going to ask the agent which of two stimuli has the smallest number of link to the target stimuli. So if the ice cream is this green node and the basket is this green node, so on the learned transition, these two nodes have the same number of links to the target 3. But if our agent is able to infer the existence of this missing link using transfer, it will infer that the basket, the node over here, a smaller number of links to the target. And this is what actually this agent do. It can answer this question correctly by inferring the existence of unobserved link. So using inference of approximated transition metrics, other learning transition metrics enable inference of transition that have not yet observed. But this is not the, sorry. And so now for human, whether human can actually represent and transfer some knowledge. For that, we have this following behavioral experiment in which a participant came to the lab during 2 successive days, and they were segregated into 2 groups. One group learned hexagonal graphs on the first day, while the other groups learned a community structure graph on the first day, and both of them on the second day learned this hexagonal graph with these missing links. And of course, they never observed the bird view of the graph and had to learn it using the sum game. So although these larger, really larger, 36 node with degree 6 participants were able to learn it quite good. And when they did the task of answering which of 2 pictures are closer to the target picture, participants that experienced hexagonal graphs on the day before did this task much better than, significantly better than participants that experienced the other graph. And they were able to answer the inference questions with this missing, by inferring this unobserved link significantly better than the one with the wrong structural prior. So it means that humans use prior knowledge to infer missing links, and they can represent transition metrics abstractly and transfer this knowledge to complete unobserved transition. But this is not the only thing that you can do with structural knowledge. Now look on this community structure graph. You see that there are that the blue nodes here are quite special. So there are structures or structural forms in which you have some kind of special node. And with this basis set, you can, in this basis set, for example, what you do is that you have 2 types of basis set. One that tells you which nodes connect, belong to which community, but you also have a basis set that tell you which node are the connecting node. And the hypothesis is that these basis sets are being learned during our development, during our life, and therefore we experience these connecting nodes in different circumstances. This allows us also to presumably generalize this knowledge and understand what is the semantic meaning, or what is the meaning of what can I do with this connecting node. So now when I use this basis set and approximate the transition and learn the emission metrics, this directly leads to the inference of the connecting node when the semantic meaning is already known. So here again, the model can infer the structural form, the size of the graph, and also infer correctly the identity of these connecting nodes. So basis set representation allows direct inference of imported nodes without the need for further calculation. And for the behavioral experiment, it's very similar to what we did before, but now on the second day, all participants learned this community structure graph. So when they learned association, they determined their own pace. They could determine how long they to stay on this on each association. And we see that participants that learned the community structure the day before spend less time on learning each association. So they learn faster. And not only they learn faster, they also perform the task better, a navigation task, in which they start with one picture and had to choose between two other pictures in order to get as fast as they can, with the smallest number of steps to the target picture. They repeat it until they get to the target pictures. And you see that participants with the correct prior that expect to see this community structure perform this task with much less steps. So this kind of suggests that structural prior leads to a different learning and behavioral policy. So what is this policy? So if we look on all occasions in which participants had to choose between these two nodes, that one of them is a connecting node, we see that participants that expect to see this community structure prior tend to use a connecting node much more. And they did it even though it was the wrong answer. It led them to far away from the target. So this was kind of mean that they come with this policy which I want to choose connecting node. So it seems that participants with the correct structural prior identify better connecting node. The focus of attention during learning and navigation is affected by the prior expectation of a structural form. And inference of structural form leads to behavior policy, which is beneficial for that structural form. And in a very similar fMRI experiment, we have a preliminary result that suggests that this flexible and abstract structural form is encoded in DMTL. And to summarize, structural forms can be represented in the form of basis sets, and this type of representation allows generalization over particularities of the current tasks, such as stimuli and size. It enables inference of unobserved transition and allows the use of beneficial behavioral policy. Participants with the correct structural prior can infer unobserved transition, and this implies that they can transfer the knowledge of abstract transition structure. And learning and behavioral policy is affected by prior expectation over the structural form. And preliminary fMRI results suggest that abstract and flexible representation of the structure exists in the spinal cortex or MTL. And that's it. And thanks for my supervisor, Steve Canrelay, and the member of the group. That's it.
Thank you, Shirley. I think we are right on time, so we have a few questions now. The first one is from Leonard. Great work, thanks for the talk. Unsure if I understood correctly, but does the HMM assume that transition matrix is known entirely to subjects or agents? If yes, how would this be realized in real world situations where experience?
No. Sorry.
I mean, it's a long question, so where experience of state-space transitions is rather gradual and humans might forget, how would the model account for inaccuracies resulting from mapping sequential inputs to a discrete structure?
So first of all, it's not, if I understand the question correctly, it's not that the model learned the transition structure or hold this particular transition structure. It's kind of estimated from basis set that already represented. So it exploits knowledge that already been observed. For the neural mechanism of how, so this model is not accounting for any noise. Therefore, it's doing much better. It reached 100% correct, so it's doing much better than human participants. And for how the brain can do it exactly, I can just speculate.
Got it. Yeah. So next one we have from Habiba Azab. Really cool talk. I'm wondering what you think of the case where a stimulus action or a state action association also needs to be superimposed on the abstract structure. How or where do you think this would be represented?
Well, so this is also, I can just speculate, but I kind of think that, so in my imagination, the way that I view it is that we kind of, we can use, because these structures are really prevalent, so we can use this basis also as kind of a basis set to approximate the function of how good we are in doing different actions. Okay, like let's say I want to kick a ball, and because I played a lot of time soccer, I can estimate using this representation how good am I and I can have this function of probability of success in this action of kicking a ball using this representation. But this is, just very speculative, like the way I think about it.
Yeah. Next, we have a question from one of our panelists, Leonie. Why don't you just go ahead and ask a question?
Yes, thank you. That was incredible. Thank you, Shirley. I had a question regarding the preliminary fMRI result. I was wondering how, and maybe this is not the scope of the question time, but I was wondering what analysis method you used to decode the structure in what looked like, I think, the left entorhinal cortex.
Yeah, okay, so yeah, this is quite a complicated analysis. It's not a regular analysis. So it will take me a while to explain. Yeah, I guess it's, I'll be happy to give you.
Yeah, can I quickly interrupt, because we have just about a minute left before we'll get kicked out of this room. So maybe this could be taken up offline. I'm sorry that we are running out of time right now. All right. I just wanted to thank the panelists again, and thank you for and all the attendees for stopping by. And that's about it. So before we actually get kicked out, I guess we can finish the presentation. Thank you.
All right. We are on YouTube. Okay, I'll start with the recording again. Sorry about that.
All right, looks like it's time. All right, I will get started. So welcome everyone to this session. This is room 7, and we have a really exciting lineup talk today. And before we get started, I want to remind you to click on the Q&A at the bottom of your screen and ask questions and vote for questions during the talk. And I will ask them after each speaker's talk. And so I will introduce the first speaker. Our first speaker today is Dominic Standage, and he is a Marie Curie Senior Research Fellow at the University of Birmingham, UK, and he's currently on pandemic leave at Queen's University, Canada. So Dominic uses modeling and data analysis methods to investigate the neural basis of cognitions and consciousness. And Dominic, the floor is all yours.
Thanks, Mengsen. Okay, I'm going to talk about differential modular dynamics in marmoset cortex during conscious and unconscious states. So a bare bones and perhaps cowardly definition of consciousness is that it's the ability to sense and respond to stimuli. As such, a great way to investigate consciousness is through the use of general anesthetics, which disrupt that ability temporarily. The cellular effects of general anesthetics are quite well understood, and they have to be used in medical practice. But how those cellular effects play out at the large scale level of brain networks is not so well understood. A great way to investigate those large scale brain effects or network effects in combination with anesthetics is through the use of neuroimaging, such as functional magnetic resonance imaging, fMRI. So in this study, 4 marmosets, so little New World monkeys, were scanned, were put in an MRI scanner in 2 conditions: when they were awake and when they were under the influence of the anesthetic isoflurane. In each condition, they were scanned 6 times for 20 minutes each. The rough overview of the methodology is we have 116 cortical regions that we're recording from, and we see these time series. The nodes here are brain regions under this partialization scheme. So we'll try to construct brain networks in 1-minute time windows, W1, W2, W3, and so forth. And so what we do is for each pair of regions, we calculate the covariation of their hemodynamic response and then slide the window over 20 times. What that yields is a multi-slice or temporal network, where in each time slice we can see these whole brain, or in this case, whole sort of monolithic cortical networks, where in these maps, again, the nodes are regions, and so yellow is high covariation, blue is low covariation. And we're interested in particular in the modular structure. So modularity refers to subnetworks. So we characterized by strong or dense connectivity within the modules and weak or sparse connectivity between them. So if we look at the schematic here, we've got, for each window, we have what's called a modular partition. We've got 4 color-coded modules. If you look at the red module, for example, it has 4 members, and it has 6, then it has 4, then it has 3. So there's a reconfiguration of this modular partition that goes on in time. And in recent work, including some of our own in preparation, there's evidence for the statistics of this reconfiguration correlating with performance on cognitive tasks. And so we want to ask, do these modular dynamics differ between consciousness and unconsciousness? So the first hypothesis we tested was that cortical modular reconfiguration is less coordinated during unconsciousness. So why do we hypothesize that? The short answer is that it's a prediction of an earlier study by our group where we looked at similar measures of modular dynamics from across depths of unconsciousness, but not looking in the conscious state. A slightly longer explanation is, if you think of functional connectivity as a dynamic system riding on the underlying structural connectivity, well, if anesthetics weaken functional connectivity, and there's evidence that they do, and if that weakening renders the functional connectivity sort of just more similar to it, sort of sinks into the underlying structural connectivity, it impoverishes the dynamics, and so the dynamics don't support groups of brain regions forming modules together in a coordinated manner. And so the first measure that we take in relation to this hypothesis is referred to as disjointedness. So it's simply the number of times for each region that region changes modules by itself. So at the time it changes, no other region changes with it. And we divide that by the number of transitions between windows and average over the regions. And so in the awake condition here in the dark shade, these are the within subject means. So for each monkey across all the scans, this measured disjointedness, we see it in the awake condition, the anesthetic condition, and the light shade. And you can see that for each monkey, we have these pairwise increases in disjointedness, this haphazard, uncoordinated reconfiguration. And the dashed line just draws your attention to the fact that all of the anesthetic data are separate from all of the awake data. So it's sort of an intuitive, if not formal, inverse of this measure is referred to as cohesion strength. So it's the number of times for each region that it changes modules together with other regions. And that's relative to the number of transitions, and it's weighted by the number of regions it changes with. And so, this captures coordinated reconfiguration. We see that in the awake state, we have higher cohesion strength than in the anesthetic, than under anesthetic. So far 2 for 2. Sorry, 1 for 1 in terms of hypotheses. So the second hypothesis we tested is that cortical networks become fragmented during unconsciousness. And the long and the short of it is there's a fairly large body of data that characterizes unconsciousness this way. Modularity is fragmentation. It's subsectioning of networks within larger networks. And so, yeah, we can count the number of modules that we identify and have them over the scans, and we see that we have more modules. And so greater fragmentation in the anesthetic or under anesthetic. So next hypothesis is that, and I can't see it here, fragmentation is characterized by hierarchical network disintegration. All right, so there's a little bit more to unpack here. Well, first thing we need to do is to identify subnetworks across these modular partitions. As the modules ebb and flow, we want to nail down summary structure. And so we simply calculate the probability across all windows, scans, and subjects that each pair of regions was together in the same module. So that's referred to as a module allegiance matrix. We calculate that matrix in the await condition. So here, we line up our regions on the axes. And so, yellow means that, these two regions tended to be together, and blue means they tended to be in different modules. So we can then cluster that network, or that allegiance network, grouping like with like, and we see we have significant structure. So this is effectively, these are sort of the modules or subnetworks from which the reconfiguration occurred. It's a nice summary. And so we can project these subnetworks onto brain plots, and we see that we have sensible networks that are, we have a visual frontoparietal network, ventrotemporal prefrontal network, and the somatomotor network. The gray blobs in here are subcortical areas that we didn't record from. So fragmentation is characterized by hierarchical network disintegration. So now we can so we've got our clustered module allegiance matrix with our identified networks. We can now we take the same clustering solution and impose it on the module allegiance matrix in the anesthetic condition. So if you look, you can sort of eyeball this, that you can see the networks are, look at network 2, it seems to be disintegrating. Network 3 seems to be disintegrating. And as they disintegrate, they sort of smear into one another. So this is the disintegration that we're talking about. We can measure that, we can quantify that observation with a measure within network integration. It is effectively the sum of regional allegiances. So summing up these weights relative to network size. We see that, in network 1, 3 out of 4 networks show this within network integration, it drops off so that they're disintegrating, not in the 4th monkey. So it's not totally clear. But if we look at networks 2 and 3, you can see that they're disintegrating, and that's borne out in this measure within network integration. So we now want to look at the smearing between them. We're going to calculate a measure called between network integration, which is effectively the sum of cross allegiances. So you put networks 1 and 2 here. These are their cross allegiances. That's relative to their within network integration. And so no, networks 1 and 2 relative to their, let's call it their baseline, they're not smearing into one another. But clearly networks 1 and 3, they absolutely do begin to smear. You can see it goes from dark blue to lighter blue. We see the same thing between networks 2 and 3. So it's happening in most of the networks, but not all. And so finally, we want to address this hierarchical aspect of the disintegration, the fragmentation. And so the 3 networks that we've derived from the modular partitions, one is a somatomotor network. Hierarchically, this is low level. Thanks, Ming-Seng. The ventrotemporal prefrontal network is high level. This is a pure association network. This visual frontoparietal network is kind of mixed low-high. So one of these networks did not disintegrate clearly. It did not smear into the others uniformly. And we'd expect that to be the somatomotor network, but it wasn't. It was the visual frontoparietal network. So that's somewhat surprising, and particularly in relation to our previous work, where we sort of had a very clear hierarchical delineation of these measures. So perhaps that suggests that there's a difference in terms of hierarchical fragmentation from consciousness to unconsciousness, as opposed to from shallow unconsciousness to deep unconsciousness. And what we want to do next is sort of dive into the hierarchical structure within the networks. And you can see that there is still some structure here, despite this disintegration that's going on. So I'll wrap up now. Collaborators on this project are Jason Gallivan at Queen's University, Stefan Everling, Yuki Hori, and Ravi Menon at Western University, both universities in Canada. These data were recorded by Yuki in Stefan's lab in collaboration with Ravi, were published earlier this year in Cerebral Cortex, and they were kind enough to share them with Jason and me. Thank you. If you have any questions or comments, there's my email address.
Thank you so much for the very interesting talk. We will wait for people to ask questions. Please post your question to the Q&A session and I will ask them. But while we're waiting, I would ask you something. This is actually very interesting to me. One thing I'm wondering about is whether in EEG, I know that there is a big difference between the sleep and anesthesiology stage. So is there this similar kind of, is the cortical network reconfigured differently during regular sleep rather than?
Right. I don't have a definitive answer. I'm actually quite new to consciousness, unconsciousness, as a researcher, not as a human being. Yeah, I mean, principally, you know, this approach to network neuroscience in the area of consciousness and unconsciousness, you know, you're looking at sleep studies, you're looking at anesthetic studies, and some clinical studies with patients in persistent vegetative states and unresponsive states. Yeah, my impression is that there's a lot that's in common, but there's a lot that differs. In terms of the hierarchical aspects of these things, I'm pretty sure that in sleep studies, you sort of get, you know, there's psychophysical evidence for responsiveness that you wouldn't get under high doses of anesthetic. And this sort of hierarchical breakdown, you know, tends to, at lower doses of anesthetics, you get the higher areas of brain hierarchy sort of begin to fragment first, and at higher doses, it sort of cascades down. I think that is generally the case in sort of depths of sleep as well, that probably aren't as easy to categorize. But I'm pretty sure that there's the retained responsiveness is there in sleep, more so than it would be under anesthesia. But yeah, that's a really good question. I do need to know this literature better, but I mean, yeah, I'm enjoying it.
We have a question coming in. Has cortical modular reconfiguration been used in other studies as well to characterize different states of consciousness?
So static modularity has been, I know has been looked at in some anesthetic studies where you take one modular configuration. In terms of dynamic modular reconfiguration, only our study, to the best of my knowledge, earlier this year with macaque monkeys, also in collaboration with Stefan Everling's group, and working with Jason Galvan and I, were working with a data set that they were published years ago. And yeah, we looked at, you know, dynamic reconfiguration across depths of unconsciousness. They had 6 dose levels of isoflurane. I'm unaware of any other dynamic modular analyses of unconsciousness.
Thank you so much. We are going to move on to our next speaker. Our next speaker is Vicente Alba Suarez, and he will set up his slides now. He's a first-year master's student at York University. So the case study he will be presenting today is a continuation from his undergraduate honor thesis to his master's work. And the work is interdisciplinary in nature. He explores cognitive neuroscience and the connection with genetics. So Vicente, the floor is all yours.
Thank you so much for that introduction, Mengsen, very kind. And I'm really excited to share with you my work today as I take you into this relatively new space of the unknown. But let's get started. So the first thing I'd like to talk to you about today is human brain-derived neurotrophic factor, which is a key brain protein responsible for a wide variety of functions. But the most important ones that I'd like to mention are dendritic maintenance and synaptic plasticity. So we can begin to see how this is a protein responsible not only for cellular level connections between two cells, but also at a network level in creating connections between regions that functionally co-activate as a result of certain stimuli. With that in mind, I'd like to draw your attention to the genetic code for BDNF, which is outlined here, and specifically this region over here is subject to a well-studied mutation known as the Val66 MET polymorphism. And what happens here is that the G nucleotide outlined right here changes to an A. And what happens is that there's a full switch in amino acids from valine to methionine. And that's critically important because this mutation and SNP has been studied across a wide variety of disciplines in neuroscience. I'm going to outline just a couple of the most relevant ones to my research. First, at the cellular level, we can compare val-BDNF and met-BDNF, specifically taking a look at the number of dendrites and the length of them. At a cell that expresses met-BDNF, the dendrites are, for one, shorter, and there are a fewer number of them in comparison to a cell expressing val-BDNF. Then, moving on to a network level, we can also take a look at differences in functional connectivity or the strength of connection between different regions, simply based on the functional connectivity of like structures such as the posterior cingulate cortex, the medial prefrontal cortex, the inferior parietal lobule, and these are all within the default mode network, which is a critical brain network that has been involved in internal cognition and visual attention. So these are some really interesting results considering what we just saw in the cell, but I'm going to talk about this a little bit more later. And then even in the field of non-invasive brain stimulation, the polymorphism for Val66 MET has been shown to influence the responsiveness of different brain stimulation protocols. So when researchers used CTBS and ITBS, which are two different repetitive transcranial magnetic stimulation or non-invasive brain stimulation protocols, when they used it on the primary motor system, it was found that individuals who were MET homozygotes or MET carriers, meaning they had a copy of the MET allele, actually showed reduced responsiveness overall to brain stimulation. Now, you might be asking yourself, why does any of this matter? Well, let's start from the beginning. At the cellular level, we can see impacts in dendritic outgrowths and the formation of synapses, which then leads to differences at the network level, or at least we hypothesize that it does, because we observe these differences in functional connectivity, and it also brings up a really important question of how does this affect normal brain functioning? And even beyond this, we're seeing effects with non-invasive brain stimulation. And it really calls into the question the viability of different neurointerventions like RTMS, because a lot of academic and clinical research is currently applying neurointerventions like TMS to try to modulate connections and signaling in brain networks, like in the default mode network that I outlined earlier. So with all of this in mind, I set out with a couple of goals. My primary goal was to establish whether the BDNF polymorphism for Val66 MET has the capacity to influence TMS, the effectiveness of TMS in critical brain networks. And the brain networks we studied are the default mode network, the dorsal attention network, and the frontal parietal control network, which are key networks that we're studying in my lab, and they're all involved in or have a link to visual attention. Furthermore, we wanted to also confirm past studies that have showed reduced functional connectivity in networks like the DMN, the default mode network, for MET carriers and homozygotes. The way we went about this is doing a case study. So we acquired one Val homozygous and one MET homozygous participant. Both were young adults that self-reported no medical conditions and were tested for cognitive deficiencies and depression. So the way we went about doing this, first we performed a resting state functional MRI scan just to acquire baseline measurements of functional connectivity for each participant. We then subjected the participants to ITBS and CTBS. So ITBS is intermittent theta burst stimulation, and CTBS is continuous theta burst stimulation. And these are two RTMS protocols that produce opposing effects on the network that we were stimulating, which was the default mode network, the DMN. And specifically, we stimulated the left IPL that you can see right here, or the left inferior parietal lobule, which is part of the default mode network. And directly following brain stimulation, we administered another resting state fMRI scan as we wanted to evaluate the causal changes in network connectivity before and after brain stimulation. The other really important part of our study was genotyping, which we did through the collection of a saliva sample and through PCR and restriction endonuclease analysis. So before I get into my results, I'd like to just share a couple of predictions with you. For the Val homozygous participant, we expected that there would be typical functional connectivity in the DMN before any brain stimulation occurred. And then with the ITBS brain stimulation, we expected a decrease in functional connectivity within the DMN, but an increase in functional connectivity between other networks like the dorsal attention network and frontal parietal control network. For the CTBS condition, we expected a decrease in functional connectivity within the DMN, and we expected an increase between the DMN and other networks. And importantly, these are all measurements that we have observed in past studies. For the MET homozygote, we expected atypical functional connectivity from the start before any brain stimulation. And we also predicted that there would be minimal changes in functional connectivity before and after both brain stimulation protocols. So the first thing I'd like to show you is actually just a visual representation of correlations between structures in the network of interest, the DMN. And if we take a look at the correlations between the Val homozygote and the MET homozygote, we'll see that the strength of connectivity is lower for the MET homozygous participant in key areas like in the PCC, in the posterior cingulate cortex, and in the medial prefrontal cortex, where there seems to be little to no correlations. So this is incredibly interesting, but it does reflect previous studies and what they have shown in MET homozygous and MET carriers individuals. Moving on to the brain stimulation results, I'd like to just briefly go over the graphs here. On the y-axis, we have the Pearson correlation coefficient specifically for the stimulation site, so the left IPL, and we're comparing those correlations to other network structures, both within our network of interest, the default mode network, and between our other networks, the dorsal attention network and frontal parietal control network. What I'd like to highlight here is that for the Val homozygous participant, we observed expected results. With CTBS, we expected an increase in activity or in correlation within the DMN, and we did observe that. And we expected a decrease in correlation between networks, and that's what we observed. For ITBS, similarly, we expected a decrease within network, and we observed that with the exception of the MPFC. And we did observe increases in correlation at the DAN. Importantly, these results reflect typical network connectivity, and there's typical network coupling in here in these results. Moving on to the MET homozygous individual, this individual showed a mixed results, particularly when we take a look at the before and after brain stimulation, we'll see that there's a mixture of increased and decreased correlations between the stimulation site, the IPL, and other structures. So while in one structure, it may have decreased the correlation, in another structure within the same network, it may have increased. So that was incredibly interesting to see. And another thing I'd like to point out is that for this participant, there is a positive correlation of the frontal parietal control network and the default mode network, which is not something we typically observe in resting state. So that was something really interesting and something I'll bring up a little bit later. But now I'd like to just quickly give you an overview of the magnitude of changes for the two individuals. So as it turns out, what we see for the Val homozygous individual is that when we stimulate their brain with ITBS or CTBS, the magnitude of change in terms of correlation strength is larger for these individuals in comparison to the met homozygotes. And that's particularly evident in the ITBS condition, not as much in the CTBS condition, and we're interested in taking a look at why that might be the case. But anyways, if you take anything away from what I've said today, it's that before brain stimulation, connectivity in the met homozygote is abnormal in comparison to the val homozygote. And if following brain stimulation, the val homozygotes have overall stronger responsiveness to brain stimulation. Now I'd like to just really quickly go over why what might be happening at the cellular level that might be leading to these network effects. And so this is my hypothesis as to what might be happening, and more research is required in this area. Basically, I'm thinking that there might be an error in protein folding that results in key proteins not being able to bind to the structures of BDNF. So for example, carboxypeptidase E might not be able to bind to pro-BDNF and move it outside of the cell. Or convertase enzyme might not be able to bind to pro-BDNF and cleave it into a mature protein. And that's particularly concerning because then it would never be functionally active and it would never induce cell level changes. So beyond this, we have a couple of important questions, but I'm running a little bit low on time. So briefly, I'll just like to say that we really are interested in looking at how structures might be impacted by the MET allele and having a copy of the MET allele. Like we've already observed differences in episodic memory at the hippocampal level. We observed this strange coupling between the DMN and the FPCN, and this is abnormal because of the genotype. So we were wondering if the genotype does actually have an influence on network coupling. And for the future, we'd really like to increase our sample size to increase generalizability. We'd like to stimulate other brain networks to see if this is reproducible. And overall, we'd also like to study the brain behavior relationship and how the gene affects the brain. So with that, I'd like to thank everyone for their attention and for listening. And thank you to NeuroMuch for hosting me in this talk. Thank you so much.
Thank you so much for the talk. It's very interesting. Let's wait for some questions. Please don't be shy and post questions to the Q&A. While we're waiting, I will probably chat a little bit with you. So this is very interesting for me. I'm wondering if you are, especially for the last part, are you thinking about using any pharmacological interventions because you're talking about molecular level? Is there any plan to be able to intervene and have a controlled comparison?
That's incredibly interesting, and it's something we hadn't fully considered yet, just simply due to the nature of, you know, we've had a lot of obstacles in the way to get things rolling. One of the biggest one being COVID-19, but before that, there were a couple of obstacles along the way. So right now, what we were interested in this study is highlighting, okay, are we going to observe changes when we do brain stimulation between individuals that are met homozygous or val homozygous? And so that's ideally what we wanted to observe, at least for the first study. But now we might consider actually seeing what pharmacological agents how might they might impact network connectivity and cellular level differences, because we're incredibly interested in seeing, you know, will this have a broader overall effect in brain stimulation studies and non-invasive brain stimulation? So we'll look into that. I think that's a really good question and a really good point. Yeah.
Very nice. Thank you. I want to ask you more questions, but our schedule is ticking. So I will move on to the next speaker, and I will introduce him while he's setting up his slides. So our next speaker is Scott Squires. He's a neuroscience PhD student at the Center for Neuroscience Studies at Queen's University. So Scott studies the basics and clinical neuroscience of rumination, which is defined as the mental state of repetitive cyclical thinking about causes and consequences of distressing feelings, unfavorable experience and stressors, and unachieved goals. So, Scott, I'm very excited about your talk. Please take it away. You are muted, I think.
Okay, there we go. Thank you very much for the introduction. So today I'm going to be talking about mentation rate during resting state fMRI and trait depressive rumination. Is there a difference between depressed and never depressed individuals? Before I get into that, I just wanted to use this opportunity to plug this research network that I'm a trainee for, which is the Canadian Biomarker Integration Network for Depression. So this is a multi-site research network first developed and kind of coordinated by Sydney Kennedy, a psychiatrist with the University Health Network, with the goal of trying to find biomarkers for depression using a machine learning approach, combining clinical data, blood data, brain scans, and so on, and then trying to find individualized treatments for people based off of these data. There's a multitude of studies. We're now on to study number 15 from the CanBind initiative, but today I'm going to be talking about CanBind 4, which was collected at Queen's University. So I just wanted to also show this picture because it just shows how big the group's gotten. This was taken in Toronto a few years ago. I'm all the way here in the back, right here. So, and Sydney Kenny's a little bit here. So that's kind of just like a bit of an analogy of kind of how far separated I am as a trainee from the top kind of levels of this organization, but it's been a lot of fun. Okay, I can't. Sorry. There we go. Okay. So major depressive disorder is characterized by depressed mood, anhedonia, weight loss or weight gain, insomnia or hypersomnia, psychomotor agitation or psychomotor retardation, fatigue, feelings of worthlessness or excessive inappropriate guilt, concentration difficulties or indecisiveness, and then recurrent thoughts of death, suicide, or suicide attempts. You need 5 out of 9 of these symptoms, and one of the first two, you have to endorse one of the first two in order to get a diagnosis of major depressive disorder. And this disorder is clearly debilitating to the individual, but it also creates problems with academic and career functioning, interpersonal difficulties, health-related problems, and financial difficulties, both for the individual, their family, and society as a whole. What I'm interested in studying, as was mentioned in the introduction, is cognitive rumination, or specifically in this case, depressive rumination, which is a mode of responding to distress that involves repetitively and passively focusing on symptoms of distress and on the possible causes and consequences of these symptoms. And the idea here is that by constantly dwelling and cyclically thinking about negative experiences, it increases negative affect, reduces motivation to complete instrumental tasks, it impairs problem solving and creates friction in social relationships. And these factors all combine together to increase the risk for depression. Depressive rumination as defined by the response styles theory is commonly measured with the ruminative responses scale of the response styles questionnaire. This was originally developed in 1991 as a 22-item version, but the instructions are still the same today. So it's a 1 to 4 Likert scale, and basically participants are asked to respond to a bunch of general statements related that covers how they respond to whenever they feel down, sad, or depressed. And this measure was first made to kind of show the people's responses to the negative feelings of experiencing a very devastating earthquake in the San Francisco area in the late 80s. There were multiple criticisms for this study, or this questionnaire, mainly the confounding depression symptoms with depressive rumination. So a 10-item version was created with 2 factors. Brooding is defined as negative aspects of self-reflection, focusing on abstract why me kind of issues and obstacles that to overcoming problems. So this could be like thinking, why am I deserving this? Thinking about recent situations, wishing they had gone better. Why can't I handle things better? And then there's reflection or pondering, which is emotionally neutral or positive aspects of self-reflection, focusing on understanding oneself and situations on problems. And these can be like responding to, I analyze recent events and try to understand why I'm depressed. I go someplace alone to think about my feelings, and I write down what I'm thinking and analyze it. So when thinking about rumination, a common issue where people who experience high rumination is that it's a repetitive and recurrent thought process that's difficult to disengage from. And when understanding kind of the processes of rumination, it's important to define what a thought actually is. So a thought is a transient mental state or a sequence of mental states that are cognitive and or emotional and can be described in terms of their contents and the relation that a person has with those contents. So these can be everything from perceptions, beliefs, fears, imaginings, memories, and so on. And because thoughts are defined in this way, in terms of their contents, that means that a change in thought could ostensibly be measured objectively as a change in ongoing categorical representations encoded within neuroimaging data. However, instead of defining a priori which categorical representations are being adjusted as a function of rumination, one thing what we can do instead is it is possible to track holistic changes in semantic transitions by examining whole brain network activity transitions. And the rate at which these network activity transitions can serve as a measure of mentation rate, also known as thought turnover rate. Now, in calculating mentation rate, it leverages the 15 network templates created by the Human Connectome project. So this was created in 2013 by using 812 healthy young adults that sat and did a 15-minute resting state scan, and then group independent component analysis was ran on the resting state data to find 15 spatial networks. These spatial networks will be useful for defining the thought transitions, as I'll describe in a second. So to take these 15 networks and reduce them down to a dimensional space that's easier to work with, we implemented a technique known as T-distributed stochastic neighbor embedding, which is a nonlinear dimensionality reduction technique for embedding high dimensional data into either 2 or 3 dimensional space, but in this case we did 2 dimensions. And when applied to the time series of resting state fMRI data, it looks something like this, where if you can imagine on the left, your 15 network representations, so it's in 15 dimensions, not 2 as is shown there, is moving around in this 15-dimensional network space during rest. And at every time point, so every TR, every volume, I should say, it's creating a point in this 2-dimensional space. So if there's not much connectivity or connections within the content that you're thinking, they'll show up as disparate points. But if there is a thought that someone is holding on to for a long period of time, it serves as an attractor within this network space. And you can see that they start chaining together every volume to create a metastable time point. So here's an example from the study that validated this technique from members of my lab. And this is an example of a 2-dimensional representation of the 15 network space. And in yellow is the beginning of the scan, and in blue is the end of the scan. So every TR, there's a new dot added to the this grid here. The axes don't really matter because it's a multi-dimensional space, but the idea here is that as they cluster together into these chains, they form metastable time points, which are separated by metastate transitions as person engages in a different thought. So this is validated using a movie paradigm where using the Human Connectome project data set, participants were just passively watching a movie, and from time point to time point, step distance vectors were calculated, and essentially in this 2-dimensional space, rapid changes in the step distance vector shows up as a spike, and it happens to salient things in this movie paradigm. So whenever there was a scene change, or there was an event that happened within the scene, or there was a camera cut, or something new was added to the information, or there was something visually changing, this could be decoded from this 2-dimensional network space. So using this technique, I was interested in figuring out, is trait rumination, both brooding and reflection, predictive of mentation rate at rest? And does this depend on whether the individual is depressed? So 88 depressed and 63 never depressed from the Canbind-4 study were used for this analysis, and the ruminative responses scale was used to measure rumination. Mentation rate was calculated as I was just describing. So it was a 10-minute resting state scan, and then using FSL's dual regression technique, those 15 networks from the Human Connectome project were regressed back onto the subject level data. And then TSNE was done to reduce the data. This was done 100 times, and the average was calculated for the mean step distance vectors. And then to detect the metastate transitions, Mahalanobis distances and then 2D network space was calculated across those time points, and a peak finding algorithm was used on the step distance vectors to identify the transition time points that occur at the boundaries of metastable time points. 0.06 prominence threshold was used to define the metastate transitions, and then vectors were inverted, and the peak width of 10 with 10 continuous levels that aren't in transition was defined as the metastable time points. These were then divided by the time series to get the mentation rate per minute. Multiple linear regression was done to basically look at brooding and reflection as predicting these mentation rates, controlling for agent sex. So looking at main effects, both reflection and brooding were higher in the depressed group, but there was no significant difference in mentation rate. But looking at this in the multiple regression model, it was statistically significant, accounting for almost 11 or a bit more than 11% of the variance in the network transition rate. And looking at the group results, the same thing as I just showed you, there was no difference even with these other factors in the model. And with reflection, there was no significant association with mentation rates. But interestingly, with brooding, what we see is a statistically significant result where each point on the brooding subscale was associated with 0.09 fewer network transitions per minute. So basically, the more that people self-reported brooding on average, the more at rest they were reducing, or at rest they were having fewer thought transitions, essentially. So in conclusion, trait depressive rumination that is characterized by brooding but not reflection is associated with a slower resting mentation rate, whether someone is currently depressed or has never experienced an episode of depression before. A major limitation of the study is the fact that they were doing a resting state scan. They weren't ruminating in the scanner. So, or they might have been, but we weren't inducing them to ruminate in the scanner. So future directions will investigate whether this association indexes the repetitive recurrent nature of brooding by assessing state rumination within an fMRI rumination induction task. Thank you for listening.
Thank you very much. This is a very interesting talk. And let's see if anyone has asked questions. I strongly encourage people to ask questions. While we're waiting, I'm going to ask a little bit. This is very interesting to me because I also do nonlinear dimensionality reduction with the fMRI and beyond. So Tessle is a little bit, has some stochastic components in it. And I'm wondering if, as you're changing some of the, if you change the dimensionality of the embedding, or you're running multiple different, I think there's also like perplexity or something like that in the, if you change this parameter, how much would that affect your result?
Okay, so I didn't manipulate perplexity to see how it affected the results, because the previous study that I presented at the beginning that validated this technique on the fMRI time series, sorry, I should say they, the co-authors on this project, tested had no effect on this technique, but it is something that I am going to do for this data set as well before I prepare these results for publication.
Very nice. Let's see. Well, only I have the best questions. So I think we're about the time. I would love to talk to you guys individually about research. I'm very fascinated by your research, and thank you so much for giving the talks today. And thanks for the participants, attendees for asking questions. And we will end here today, and I hope to see you around at Neuromatch, and hope you enjoy the rest of the conference.
Thanks, Max, and you were a fantastic MC. So yeah, thanks again.
Thank you.
Thank you very much.
Thank you so much, Maxing.
Have a good one.
All of the research.
You too.
Cheers.
Okay. Hello, welcome everybody to this session. We have three amazing fellow and researcher today from the Einstein Lab. I'm Bruce Bear, postdoc researcher at Coron's Lab at UPenn, and I'm very delighted to have these three amazing people with me today. This is a traditional talk, so we have 12 minutes almost for each speaker to talk, and then we have Q&A that you can write it down either in chat or in the Q&A button, or just learn that you can write down in the YouTube if you are streaming, and we will copy and paste it here. Thank you again for joining us today, and without further ado, I am going to introduce our first speaker. Shivi is a fourth-year PhD student majoring in biomedical engineering at the University of South California, working with Berger's lab. He's using computational machine learning models for decoding memory information from spikes. His title for today is a double-layer multi-resolution classification model for decoding spatiotemporal patterns of spikes. Thank you so much, Shivi, and we are looking forward to your talk.
Thank you. Thank you.
Let me share the screen. Good?
Yes, that's
okay. Let me start. Hi, this is Xu and welcome to my presentation. My topic is a double-layer multi-resolution classification model for decoding spatial temporal pattern of spikes, and I'm from USC. The decoding of spike is a fundamental problem in neuroscience and neuroengineering. It can allow us to understand the relationship between neural activities and external behaviors, and it can be also used to identify the functional connectivities between brain regions. It will be also helpful to develop neural code-based processes for restoring many functions. And the model for decoding of spike can be formulated computationally as building an input-output model. It takes the spatial temporal pattern or features of spike as input and signals to be decoded as output. Depends on the characteristic of output signal. It can be a regression model if the output are continuous variable, and a classification model if it's a discrete output. And as we titled, we built a classification model for decoding behavior information Y based on spatial temporal pattern X within a certain time window around a trial event I. So it can be mathematically expressed by this equation, and if we take a probabilistic view, it can be to estimate the probability of events I given the response of X. And if we further narrow down into a binary classification problem, the classification rule is just simply set a 0.5 as threshold, then make classification. However, several challenges need to be addressed before achieving such a neural classification model. In typical machine learning method, we want the model structure like this, that we have enough sample and relatively shorter feature length so that our model get well trained and we have small number of open coefficients to estimate. However, in neuroscience and in neuroengineering, the situation we usually face is like this. The first issue arises from the high dimensionality. For the input, we have very high dimensional input space. With the development of modern recording techniques, we can record a large number of neurons simultaneously. And as we know that every neuron files with very high temporal resolution. Therefore, the feature length, which is equal to the product of number of neurons and the number of possible spike timing, is very extraordinarily high. And what's more, that we don't know what exactly time scale we need to consider the spike signals. That is, the optimal temporal resolution is unknown. I will introduce this further just in a minute. And on the output side, that the output space could be also high dimensional. Imagine, like, take the visual memory as the example. We can use numerous category or features to describe a certain image as representations of visual memories. And we don't know the optimal number of Q here to express the output features. And what's more, because we're doing classification model, comparing to the regression model, which can use continuous time steps to obtain enough samples, classification model usually use discrete data epochs. That is, you need many data points to add for a single trial estimation, and that the sample size will be smaller comparing to the regression model. And due to the limitation of human study, human subjects can at most perform like hundreds of trials in a single session of experiments. Therefore, we got very limited sample size. So high dimensionality pairs with limited sample size. This decoding estimation is really a highly underdetermined problem that cannot be solved by traditional estimation methods. Therefore, as solution, we proposed a double-layer multi-resolution spike decoding model. The model used B-spline basic function to reduce the feature length. And we have an online survey system. We ask thousands of normal volunteers to give labels to their memory image and thus reduce the output space. And also we use L1 regularized logistic regression classifier to achieve sparse model estimation. And we took advantage of ensemble learning. We have multiple temporal resolution B-spline bases, and we have an ensemble classifier to take all the temporal resolution together to make a combined prediction as the output signals. I will introduce every part in the following slides. And here is what we do for the input signals through multi-resolution feature extraction. As we know that the precise spike timing are crucial in neural decoding. And however, the optimal temporal resolution of spike is not known as a priori. If we model the spike signal with resolution that is too high, it may not sufficiently reduce the feature dimension. If the resolution is too low, we will lose the temporal structure of spike patterns. Therefore, here we use multiple classifier with different number of B-spline nodes. Different nodes refers to different resolution. A different B-spline can yield a feature vector with different time precision, that is to map the different temporal resolution of neurons. Therefore, we have such multiple classifiers to include a wide range of temporal resolution, and we will have an ensembled one to take all the temporal resolution together and make final prediction. And as for the output side, here is our experimental paradigm. We designed a memory-dependent behavior test named delayed match-to-sample test. There are 3 phases in the DMS test. In the focus phase, the patient need to click on the focus ring to tell the program that he is ready for the trial. And then he enters the sample phase. A sample image will show up randomly at this 9 position of this ring, and he needs to click on this sample image to tell that he has remembered it. And in the match phase, he will need to pick a sample image up among other destructors. And all the image we use in DMS test were labeled by thousands of normal volunteers through a survey system. And we have 27 non-mutually exclusive binary memory label to describe those images. For example, this image will be labeled as a building image, and this image will be labeled as a plant image. So here we use the memory label of the image in sample phase as our decoding target. And this is the model structure of our of the double layer model. Take the example like we use the animal and memory label animal as decoding target. We will include all the neural response of all the trials together, and we will make a Bayesian partition to obtain enough sample replicates for the cross-validation process, and also it will reduce the model variance. And there are two layers in our model. In the first layer, as we introduced, there are multiple first layer classifiers to include a wide range of temporal resolution. And each classifier will make their own predictions, and the output probability will be combined by the second layer classifier. And this second layer classifier serves like a meta-learner to make the final prediction. In this example, he will classify whether a trial contains sample image that is labeled with animal image or not. And we test our model with synthetic data first. We simulate spatial temporal features of spikes with various temporal resolutions. We simulate 5 Hz with 5 Hz baseline fire rate plus a Gaussian waveform with different standard deviations. For example, in this video, the red line shows the probability intensity function of the simulation. This simulated neuron shows a large standard deviation that refers to low temporal resolutions. And here shows a very sharp pattern. That is, the small standard deviation refers to high temporal resolution of firing patterns. And the location of the Gaussian bumps refers to the region of interest. That is, the simulated spike patterns shows a certain high firing patterns to the decoding target. And in addition to the simulation, we use the Matthews correlation coefficient to measure how accurate our model can make predictions. And the main result we can show here, like we can see a lot of the variance resolution of B-spline can be appropriately weighted by the model coefficient to fit the regions of interest. And it is noticed that there are a lot of color. The color lines means the different resolution B-splines. There are a lot of wide B-splines, and here the curves are shows like a narrow pattern. This shows that the double layer model can utilize multiple temporal resolution B-spline to capture the neurotemporal properties. And in addition to the temporal size, we show here the first layer classifies performance along with different temporal resolution here. And we also write down the double layer performance here. We can show that the double layer model can achieve the highest MCC by combining all this first layer together. And we also test our model with real human data. We tested the model with 5 human subjects data, and here shows the double layer multi-resolution model performance would decode with 5 patients with 5 categories. And the color bar shows the average decoding MCC, and the error bar shows the standard deviation of the MCC. So we can see that we can see that all the predictions are significant because they can pass 99% t-test. And we also statistically compare the single layer, single resolution model with the double layer multi-resolution models. And we can see that the double layer model can consistently outperform the best single layer model by combining this wide range of temporal resolutions. And as a conclusion, the double layer model can successfully capture the region of interest and the temporal property as we show in the synthetic studies. And the double layer model can efficiently avoid overfitting and yield accurate predictions. The double layer model can significantly beat the best single layer model by considering the wide range of temporal resolution and combine them together. And we believe that the proposed model has important implications to development of hippocampal memory processes. Yeah, I think that's all for my presentation, and I'm happy to the question.
Thank you so much, Shiwi. We have a time for one question if comes up. Okay. Did you compare your model with other methods? I saw that you compared with single model, but like with other spike modeling?
Yes, I have a baseline that is the naive Bayes as classifier, and we compare. And actually, the naive Bayes, because it doesn't include that sparse estimation, so it's usually fair to make any prediction. Yeah.
Awesome. Great. Okay, so if there's not any question, we are in 15 minutes. We have to move to the next talk. Thank you so much, Shiwi, again. That was a great talk. Interesting model for the memory. Okay, so we are going to the second talk, Penn. Very happy to have him here. He is a PhD candidate from University of South California, the same, working with Dr. Dong Sung. And his research interests includes electrophysiology, EEG analysis, nonlinear dynamical model, and machine learning, seizure predictions. And today he's going to talk about the sparse multi-scale nonlinear autoregressive model for seizure prediction. Thank you so much, Peng. And we're ready to have your talk. Thanks. I think you have to unmute yourself.
Thank you for your introduction. I'm Peng Yu. Today I'd like to talk about my research, a sparse multi-scale nonlinear autoregressive model for seizure prediction.
Sorry, Peng, also you can like full screen your monitor. We can see the next slide if you want to.
This one, is it better?
Oh, that's great, yes.
Okay.
Thank you.
So when we are talking about seizure prediction, what we do is to continuously monitoring the EEG signals, and then we predict the probability of an upcoming seizure. So here is the probability, and then once the probability is higher than the threshold, we can warn the patient an upcoming seizure. So we define the EEG signal as predictor signal, which is an hour before seizure onset, and there's another state called interictal signal. It's like a normal state for patients with epilepsy. And we are going to use autoregressive model. So here's the conventional autoregressive model, and here's a Laguerre-Volterra nonlinear autoregressive model. And then we will talk more about those two models in a little bit. So what we are really doing is try to classify EEG signals between interictal or predictor state. So here you see on the top, we have EEG signals, and of the final state, we have either the signal is interictal signal or it is predictor signal. And then it has been shown that autoregressive model is powerful for seizure prediction. This was done by previous study here. That shows that 2 levels of model can be used for seizure prediction. And the first level of model in their study is an autoregressive model. So they use autoregressive model to model the EEG signals. And then after they finish this step, they use the coefficient of autoregressive model as features for the next level of model. And the next level of model is classification model. And then the classification model make a final prediction about whether the signal is interictal or is predictor. And there is a limitation of this conventional autoregressive model. It's that it's only for linear dynamics, and it only covers short time scale. We'll talk more about this in a little bit. So we extend our study to include long time scale and the nonlinear dynamical model. So here on the left-hand side, we have EEG signal, and then this part is autoregressive model, short time scale linear dynamic model. This part is the same as the one proposed earlier, and then we include this part. So this is the part we propose here. We want to include long time scale and the nonlinear dynamic model, and we use Laguerre-Volterra autoregressive model to do this. And in addition, we also include the domain square error of the model, which represents the unpredictability of the model. And then all these features, so the coefficient from the autoregressive model, the coefficient from the Laguerre-Volterra autoregressive model, and the domain square error from both models were used for the sparse classifier or lasso logistic regression, and then make a final prediction. So this is the outline of our structure. And then we use ROC curve or area under ROC curve to evaluate our prediction performance. So here we talk about the autoregressive model. The ideal autoregressive model you see here is very straightforward. We predict the future value based on the past values. And in this case, the conventional one is linear model. So it is a linear combination of the weighted past values, to be more specific. And then, we use Volterra autoregressive model for our study, and there's a linear part of the autoregressive model. You see they are pretty much the same. They are basically the same. But we also take into account the nonlinear part. So here you see this part, we have a signal in the past and another signal in the past. So we take into account the interaction between every pairs of past value. So here we take into the interaction, counter interaction as a nonlinear dynamics. And in these two models, they can only cover short time scale, and then we'll talk more about this later. But there's a limitation of these two kind of model. And then we have linear components, as I already mentioned. We have linear component from the autoregressive model, the coefficient from the autoregressive model, and we want to use the nonlinear component. And we also use the Lumin square to represent the unpredictability. The bottom figure shows the fifth figure shows the coefficient of autoregressive model, and this figure shows the coefficient from Volterra autoregressive model. But there's a limitation of Volterra autoregressive model is very likely to have a very high degree of model complexity. So if we want to model a signal with 40 legs, then we'll have like 860 coefficient. So to resolve this issue, we employed the Lagrange-Volterra autoregressive model. Here the bottom left shows the equation, but the idea is that we convolve the EEG signals with a Lagrange basis function, and we should use the convolved signal to reconstruct the Volterra kernel, the Volterra autoregressive model. And the advantage is that we can greatly reduce the number of coefficients. So in this case, we only have 10 coefficients for 40 legs, comparing with the 860 coefficient for 40 legs is a very good advantage. And because of this kind of lower model complexity, so we can usually cover a longer time scale model. So this is the reason if we use Lagrange-Volterra autoregressive model, we can cover longer time scale in both linear part and also nonlinear part. So to summarize this part, we use a coefficient from Lagrange-Volterra autoregressive model for our seizure prediction. And then after we got the coefficients from the Lagrange-Volterra autoregressive model or conventional autoregressive model, we need to further reduce the number of features, because we have fewer sample, but we have a lot of features. And to do this, we employed the Lasso logistic regression. Here are the details of the math equation, but the idea is that the Lasso logistic regression can select important feature from all the features and then make the final prediction. Okay, and then we use AUC to evaluate our prediction performance. Here, just quickly introduce the ideal AUC. So if we have two classes, let's say the brown one is predictor state and the green one is interdictor state. So if the distribution of both two state classes are pretty much the same, we make a ROC curve and calculate the area under the curve is around 0.5. So, and if we can discriminate between two classes, we get a higher AUC. And if we can make a perfect discrimination, then we get a AUC equals 1. So the idea is the higher the AUC, the better the prediction performance. So those are my method, and let's move on to the results. So here we show an example of interdictor signal. Here's the EEG signal, and it's interdictor state. And here's the predictor signal, predictor EEG signals. And we use the method we just talked about to model those EEG signal. And here's the EEG signals, the coefficient distribution from both the AR model. Here's the conventional AR model. We have 3 coefficients here and the Lumin square error. And we use Lagrange-Botorel automacy model. Here we have 9 coefficients here and the mean square error. And then the bottom shows here, this one is the linear Lagrange-Botorel automacy model. And the bottom one shows the nonlinear Lagrange-Botorel automacy model. An interesting thing to show here is that when we look at the second order kernel, we see that on the bottom left, there's a peak here, and they are very close to the x-axis and the y-axis. And for predictor signal, the bottom left, it's actually a valley. So you see they are very different. This one here is a valley, and this one here is a peak. So, and in this specific case, the nonlinear part contribute a lot to our final prediction. So this is patient 1. We'll look at the prediction of patient 1 later. And here we give you a general idea of how the prediction look like. And we see that generally speaking, predictor signal have a higher score than the interactor signal. So we can, in this case, we are able to classify between predictor and the inter signal. In this case, AUC is around 0.87, and the second case, the AUC is 0.99, and you see, so predictor signal have high score than interactor signal. And this table shows the all prediction performance between we use conventional automacy model only or Lagrange-Botorel automacy model or the combination of both models. And we show that the combination of both models generally yield a higher prediction performance. And here it just shows that we did the pair t-test, and we show that Lagrange-Botorel automacy model is significantly better than the conventional automacy model, and the combination of most models is significantly better than both AR model only or Lagrange-Botorel automacy model only. So to summarize my results, I show that we combine the conventional automacy model and also the Lagrange-Botorel automacy model, and by using the coefficient of most model, we can have a higher prediction performance. That would be my presentation. Thank you for your
Thank you so much, Ben. Thank you, great. That was a great talk. Yeah, feel free to write a question. That was a great model for seizure prediction, and the result was promising. I have a question for training. Actually, we have a question. Let us start with the audience question. Heidi asked that, what are the accuracies for this model on seizure prediction?
What are the accuracy for this model of seizure prediction? So to evaluate our prediction performance, we use AUC rather than accuracy. The reason we didn't use accuracy is that the seizure prediction or the IEG signal is imbalanced. So for imbalanced data, accuracy may not be the good, choice, because 95% of time patients are in the interactor state. So we can just say, oh, every signal is interactor state, so we can reach 95% accuracy. So we didn't use accuracy. Instead, we use AUC. It is better for imbalanced data. And to give you an idea of how it feel like, we got around 0.8 AUC. So to be similar to accuracy metric, it's around, you can roughly say it's around 80% accuracy. I hope this answers your question.
Yeah. Also, I want to question that, like when you were training on the data set, do you use the same patient for the train and test, or do you scramble?
Yeah, that's a good question. We actually, ultimate goal might be we can build a general model for all patients, but that can be very difficult because I didn't show it here, but the electro orientation, the electro put on the head, it's very patient specific. So because of that, for now we build a patient specific model. So every patient we use their own some of the EEG for training and the other validation part, and then we test our prediction performance on test data only. So test data is set aside before our training. And we only, yeah.
Thank you. That makes sense. Thank you so much. Thank you. That was a great talk, and we are looking forward for your future results and papers. Thank you so much.
Thank you.
Okay, so by that, let's go to the last speaker, Brian. We are very happy to have Brian today. He's MD, PhD, and completing his PhD in medical engineering, focused on neural engineering at the University of South California. I say playing all the speakers are from University of Southern California, from the same lab. We are very happy to have all of them today. His thesis consists of non-parametric system identification modeling of neural sparks transformation using deep learning and applied machine learnings to guide closed loop simulation for brain-computer interface for memory restoration. Thank you so much, Brian, and we are looking for your talk. Thanks. And this, the title is Validation of the Convolutional Neural Network and Model for Spark Transformation using Generalized Functional Additive Models. Thank you so much.
Thank you very much, Dr. Furi, for the warm introduction. And on behalf of myself and my advisor, Professor Dong Song, I want to thank everybody for taking the time to listen to our talk today with the title already introduced. So just a quick plug for a recent paper that I encourage you to look up if you find anything that I spoke about today interesting that was recently published in IEEE EMBC. In July, it's under a very similar name to the talk today, and you can find it right now in IEEE Explore if you have any interest. So we're interested in identifying the causal relationships in neural activity, and this is a very important problem in neuroscience and neuroengineering because successfully identifying these functions is necessary in order to understand how neurons transmit information between one another. So we know that neurons communicate with each other via spikes, with individual neurons receiving spike train inputs from multiple neurons while also sending spike train outputs to other neurons themselves. And any modeling approach of this activity has to account for the dependence of these output neuron spike trains on both current and past spiking activity of the input neurons. And it's also well known from many prior studies that these transformations consist of both linear functions and nonlinear functions, which we refer to here as kernels. So a lot of different modeling methods have tried to capture these kernels, including basic linear models, generalized linear models, generalized additive models, and generalized functional additive models. And I just want to quickly review the difference between them and their power, because this is how we generated our synthetic data. So a basic linear model is what we all learned in middle school, y equals mx plus b. We show it here in vector form. A generalized linear model expands on a basic linear model with relaxation of assumption of the normal distribution of error, and as well as providing a link function that can essentially allow for an arbitrary distribution in the outcome variable. And the best known example of this to most people is probably logistic regression. And then a generalized additive model expands on the generalized linear model by not modeling on the initial predictors themselves, but actually modeling on smooth functions of these predictors. And this is able to capture nonlinearity depending on the functions that you use to transform your initial predictors. And then lastly, a generalized functional additive model expands on the power of a generalized additive model by integrating the predictors with regression functions. And this can allow for even better ability to model nonlinearity in the system. So, as two of the previous presenters talked about some of the work from our lab, we've used a generalized functional additive model before, and this can expand these unknown input-output transfer functions with a set of basis functions, and this can facilitate model estimation by reducing the total number of model coefficients to be learned. And some of the past work in our lab has used a multi-input, multi-output model, which itself is a combination of multi-input, single-output models as shown here in the picture, in order to capture nonlinear dynamics. And although we found some success with this, the MIMO model itself is still a shallow model in comparison to some of the contemporary model architectures in deep learning. So I think the audience is probably very familiar that over the last 10 years or so, the convolutional neural networks have redefined state-of-the-art performance in image classification. And although a basic convolution layer itself is a linear system, you can introduce nonlinearity into the system in the form of nonlinear activation functions. And we hypothesize that a potential first step in capturing the nonlinearity in these neural spike transformations is to use a nonlinear activation function that's ubiquitous within CNNs these days, called the rectified linear unit activation. And we additionally thought that the depth of a convolutional network when compared to the MIMO model would enable it to better capture kind of complex, more sophisticated nonlinear dynamics. And then, additionally, all these other properties of a CNN that we've all kind of come to know and love, parameter sharing, translational invariance, translational equivariance. We thought this might also help to be able to recover some of the nonlinearity in these systems as well. So in order to do this, we treat two-dimensional spatial temporal patterns of spikes as binary images to try and recreate the success of CNNs in order to, model spike transformations. And this, becomes a binary classification problem using a CNN where our data is images that we obtain from input neurons over time, and our labels are a binary 0 versus 1 and an output neuron, representing whether there's absence or presence of a spike in that output neuron at a given time step. So in order to do this, we generated synthetic data in MATLAB using a generalized functional additive model, as I showed previously, in order to test the performance of a CNN to recover the probability of spiking in a single output neuron over time based on input from 16 input neurons. So we use recovery of the true model kernels that we use to generate the data process as a performance metric for the CNN model. And we developed 8 neurophysiologically inspired kernels, and these had different shapes and delays. The input signals were 16 IID Poisson processes with a 10 Hertz mean firing. And to model these 16 neurons over about 40 minutes approximately at a 500 Hertz sampling frequency, this ended up being a 1.2 million by 16 matrix for the input data. So here I just show the general form of a generalized functional additive model that we use to generate our ground truth in the output neuron. So we sum theta 0, which is representative of the mean firing rate in that output neuron, with each input neuron individually convolved with one of these neurophysiologically inspired kernel functions. So I just saw a single example here for input neuron 1 being convolved with the function on the left. And then we pass that sum to the inverse probit link function in order to map this to a range of 0 to 1, which is representative of the probability of spiking in the output neuron. So of the 16 input neurons, 8 were generated as shown here with these neurophysiologic kernel functions, and 8 were generated with kernel functions set to be zeros. And the 0th order kernel represented by theta naught there was set to negative 7.6 approximately, so that we could get around a 1 Hertz baseline firing rate in the output neuron. So we wanted to do this so there was a high level of sparsity in the output neuron, so we could kind of see how robust the CNN was to extreme class imbalance that we would typically see in human spike sorted data. So this created a time series of probability at firing in the output neuron at time t. And the ground truth labels as spikes in the output neuron were then realized with the Bernoulli probability mass function with the probability equal to the firing probability intensity function. So this eventually yielded a binary 1.2 million by 1 array as our final model output. So we then took this input data and the output labels, and they were split just into standard 0.8, 0.1, 0.1 train validation test sets, and create the input neural time series images to actually train the CNN. We used a modification of TensorFlow's time series generator utility class, which created a sliding window, which is shown here not to scale with our data, but just to give you kind of a picture of how we did this. So we used our modification of the time series generator to create this sliding window that created images of size 256 by 16, where 256 time steps from t current to t current minus 256 from the 16 input neurons were output with a binary label from the output neuron at time current. And then, subsequent images were created with the sliding window with a sampling rate of 1 and a stride of 1. So this created approximately 960,000 training images for the model. So we then developed a sequential model within Keras with a TensorFlow backend and performed manual architecture optimization with respect to the number of layers of convolution, the number of filters per layer, stride and filter side within each layer as well. And we also performed some manual optimization in terms of the proportion of dropout within a Keras dropout layer that we included and used, as previously mentioned, the rectifier linear unit activation of all the layers of convolution. So all models that we tried were compiled with a sparse categorical cross entropy with an atom optimizer and default hyperparameters. And our best performing model is shown here, which consisted of 5 convolution layers, a flattened layer, dropout layer set to 0.25, and a dense 2 layer with softmax activation and bias initialized to 0. So we trained this for 120 epochs, and the model had approximately 23,000 total trainable parameters. So we show some of our training metrics plotted here, just the loss and the accuracy versus epoch, basically just to demonstrate that our dropout layer did do a sufficient job in preventing overfitting to the training set. So we used 3 different performance metrics in the evaluation of our models. The first was a minimization of the normalized root mean squared error between the ground truth probability of firing in that output neuron and the predicted probability of firing in that output neuron. Second, we use maximization of the correlation coefficient between the ground truth probability as well as the predicted probability. And then lastly, we use the actual recovery of those known 8 neurophysiologically inspired kernels, which were used to generate the synthetic data. So as shown here, our model reduced the normalized root mean squared error between the actual probability of firing in the output neuron in the test set, as well as the predicted probability of firing in the test set to 0.0132. And, we also achieved low error by accurately estimating the true probability of firing on a global and local time scale. And the scale and the plot on the left here is just kind of zoomed in on these signals on kind of a short time scale to kind of show how the predicted probability matches the actual probability. And the plot on the right is just to show that we achieved high correlation between the ground truth probability of firing and the predicted probability with a correlation coefficient of 0.9664 here. So in order to find the recovery of the ground truth kernels used to generate the data, we wrote an impulse response function that took our CNN model and a neuron index as arguments, and we used time series generator again to generate batch inputs into this. So in this impulse response function, the first input would be an image with an impulse with a single 1 at the argument neuron index at time current within a sparse matrix. And then we create 255 subsequent images for input where the impulse was at increasing time lags for each subsequent image. So we input these images into our trained CNN and generate an intermediate output from predictions from the output from the dropout layer. And then we perform matrix multiplication of that intermediate dropout layer output with the dense layer weights for the spiking class. So as shown in the plots here, of the predicted kernels overlaid on each of those 8 ground truth kernels that we used to generate the initial data, the CNN did a very good job of recovering these ground truth kernels. And this included both on the global time scale that we show here, as well as kind of more locally as we've shown previously. So, we found that our best performing model recovered the ground truth kernels and also had very good performance with minimization of the normalized root mean squared error, as well as maximizing the correlation between the ground truth and predicted probability of spiking in our output neuron. Initially, our first attempts kind of mirrored these best-in-class image classification networks, AlexNet, LayNet, some of these, but we found that they had so many trainable parameters, it was leading to overfitting. So, for our situation, we kind of discovered that using a stride between 1 and alternating between 1 and greater than 1, but less than the filter length in different layers of the CNN provided the best results for us. So in conclusion, we found that a CNN can learn the kernels that you use to generate data in a synthetic generalized functional additive model of spike transformation between layers. And our current work is basically using this same modeling approach to try and model higher order dynamics with second order self and cross kernels, as well as applying our model to animal data. So with that, I thank you all for your time, and I'm happy to answer any questions that you guys have.
Thank you again, Brian. That was a great talk. We have 1 minute if you have a question to post it. Before the question comes up, have you looked at the sensitivity of your model to number of neurons here?
You mean the number of input neurons?
Yes. Yeah.
We have. I mean, we didn't do any type of formal modeling with that. We do know that from our past work that it does scale with the number of neurons pretty well. But no, I mean, just for these initial publications, at least we kept it fixed at 16 for now.
Great, but yeah, but the architecture is scalable. I see. It doesn't matter the number of neurons, right?
Correct. And, well, at least in the this is synthetic data, so at least in that, obviously as we apply this to animal data, that's going to likely change, but that's kind of yet to be seen for us.
Also, just for curiosity, is that like dropout changing the outcome dramatically? Change the value of the dropout?
Is what changing the value of the dropout?
Yes, yeah. So you had a dropout value in your model, right?
Right.
Yeah. Does it really change the outcome if you change the dropout values?
It did. I mean, we actually, yeah. So it did. Initially we kind of started with a standard dropout of about 0.2. And, we did do a hyperparameter search around there, I think anywhere from 0.1 up to as high as 0.5. So yeah, it really did significantly help to prevent overfitting and also would change the training time as well.
That's very interesting. I usually heard that dropout is not working as well, but that's interesting that you have seen the reverse.
Yeah, it works very well for us, actually better than, L1 or L2 regularization here.
That is very interesting. Yeah, I saw that there is another regularization. Thank you so much, Brian. That was a great talk. And by that, we have to wrap up these sessions. I thank again all the three speakers today, and a special thanks for Dr. Sang for having this great PhD students. We are looking forward for your future publication and your scientific impact. Thank you so much again. See you all virtually for the rest of the conference, probably.
Bye. Thank you.
Bye, everybody.